Okay, so genitourinary system anatomy and physiology, or genitourinary system anatomy. Good. Now, genitourinary system is also known as the urogenital system. And anytime you hear genitourinary system, it refers to the organ system that includes both the urinary system and then the reproductive system. In fact, this system is responsible for producing, storing, and eliminating urine, as well as reproduction and sexual function. Okay, so basically, genitourinary system is also known as the urogenital system. Okay, and you know from anatomy 1 that anytime we say genitourinary system, or anytime we mention of a system, we are looking at group of organs coming together to perform a specific function. Okay, now for the genitourinary system or the urogenital system, it includes and it refers to an organ system that includes both the urinary system organs and then the reproductive system. Okay, so that means that it combines both the urinary system, which includes the kidneys, the ureters, the urinary bladder, and then the urethra, and then the genital organs. So the genital organs, the male genital organs on one side, and then the female genital organs or female reproductive organs on the other side. Okay, now the urinary system. Let's now focus on the urinary system, because in this lecture we are going to concentrate on the urinary system. Then another time we will look at the reproductive system. Now, the urinary system is also known as the renal system. Okay, so anytime you hear urinary system, it's also known as the renal system. In fact, this system is the body's filtration system and also the body's excretory system or excretion system. Okay, so it plays a role in filtering the blood, and it also plays a role in excreting waste products from the body. Okay, so basically, so anytime you hear urinary system, it's just the same as the renal system. Okay, so when we say urinary system anatomy, we are just talking about renal system anatomy. And this system is the body's filtration and excretion system, responsible for removing waste products and excess fluids from the bloodstream. Okay, this system basically plays a vital role in maintaining fluid, electrolyte, and acid-base balance whilst working together to produce urine, store urine, okay, and eliminate it from the body. Okay, now anatomically and functionally, okay, these two systems are interrelated, especially when we talk of the male, when we are making reference with the males, because in the males, organs like the urethra has a reproductive function and has a urinary function. Organs like the urethra, because the urethra is a passageway for both semen and then urine. Is that okay? So it tells you that these two systems are interrelated. That is why we have a combined system, genitourinary system. Okay, good. Now, disorders of this system can affect both the urinary system and reproductive system. Okay, so if someone is having a disorder of the urinary system and it's not properly managed, okay, a complication can become so serious that it can affect the other system. Is that okay? Good. So disorders of this system can actually affect both the urinary and then reproductive health. Okay, now let's look at branches of medicine relating to genitourinary system anatomy. Branches of medicine relating to genitourinary system anatomy. Okay, now we have these branches of medicine: urology, we have nephrology, we have gynecology, and then we have andrology. Now, when we say urology is a medical and surgical specialty that focuses on diseases of the urinary tract and male reproductive organs. Okay, then we have nephrology. Nephrology is also a medical specialty that focuses on kidney functions and diseases affecting the kidneys. Then we have gynecology. Gynecology basically deals with the female reproductive system. Okay, then we have andrology. Andrology also focuses on the male reproductive system. So these are branches of medicine that relate to the genitourinary system anatomy. So we have urology, which focuses on diseases affecting the urinary tract, okay, and that affecting the male reproductive organ specifically. Then we have nephrology, which focuses on disease affecting the kidney, and then disease affecting any structure within the kidney, and then the functions of the kidney. Okay, then we have gynecology, which deals with the female reproductive system, okay, conditions affecting the female reproductive system, and then studying the female reproductive system. Then we have andrology, focuses on male reproductive system. Now we are beginning the urinary system or the renal system. So like I said earlier, the urinary system is also known as the renal system, and this system is responsible for filtering blood, removing waste products, and maintaining fluid and electrolyte balance. This system also consists of the following key organs. So these are the organs of the urinary system as displayed in the diagram on our left. So we have the right kidney, the left kidney. So you realize that we have two kidneys. And in fact, the kidneys are the primary organs of this system, the urinary system. Okay, they are responsible for filtering the blood. They produce the urine, regulate blood pressure, and maintain electrolyte and acid-base balance. Okay, that is the kidney. So you see we have two kidneys, the right kidney and then the left kidney. Okay, then we have two ureters. Okay, these are the ureters from here to come and connect to the bladder. That's the right ureter, and then we have the left ureter. Okay, so we have two kidneys and then two ureters. Then we have the urinary bladder. Okay, this is more like a hollow muscular sac that stores urine until it is ready to be excreted. In fact, it expands as it fills and signals the urge to urinate. Okay, the urinary bladder is a hollow muscular sac that stores urine until it is ready to be excreted. It expands as it fills and signals the urge to urinate. That is the urinary bladder. Okay, a hollow muscular sac that stores urine until it is ready to be excreted. It expands as it fills and signals the urge to urinate. Okay, this is the urinary bladder. This is the structure we are talking about, the urinary bladder. Then we have the urethra. Okay, the urethra is a narrow tube that carries urine from the bladder to the outside of the body during urination. Okay, in males, it also conveys semen. So for this urethra, in males, it has a dual function. It's a passageway for the urine from the bladder to the urethral orifice, and it also helps to convey semen. Is that okay? So these are basically the organs of the urinary system. We have the two kidneys, we have the ureters, we have the urinary bladder, and then we have the urethra. The two kidneys, the ureters, the urinary bladder, and then the urethra. These are the organs of the urinary system. So you can see them over here. So the two kidneys, the right kidney, the left kidney, the right ureter, the left ureter, the urinary bladder, and then the urethra. These are the organs of the urinary system. Okay, good. So now let's move on to, so over here too, we have the organs of the urinary system. So we have the two kidneys over here, the right kidney, the left kidney. We have the ureter, the right ureter, the left ureter. We have the urinary bladder, and then we have the urethra. Okay, now the kidneys. In fact, when we say the kidney, like we said earlier, the kidney is the main. So now we are going to look at these four organs of the urinary system one after the other, beginning with the kidneys. Okay, the kidney is basically a bean-shaped organ. Okay, we have two kidneys, the right kidney and then the left kidney. Okay, it's a bean-shaped organ. In terms of its size, it is approximately 10 to 12 centimeters long, 5 to 7 centimeters wide, and 2.5 centimeters thick, roughly the size of a fist. Okay, in fact, the kidney weighs, the weight of each kidney weighs about 120 to 150 grams. Each kidney weighs about 120 to 150 grams. In terms of its location, it is retroperitoneal. That is, it is located behind the peritoneum. Okay, so in terms of its location, the kidney is located behind the peritoneum. The peritoneum is more like a membrane that surrounds most of the abdominal organs. But the kidney is not within the peritoneum. It is behind it. That is why we say the kidney is a retroperitoneal organ. And you see, this feature or characteristics of the kidney is very cardinal in anatomy. Very, very significant in anatomy. Okay, now, one kidney lies on each side of the spine, and it is located between the 12 thoracic vertebrae and then the 3rd lumbar vertebrae. Okay, this is also a very significant landmark in anatomy in terms of its relationship with the spine. That is for the location. Okay, it is located between the 12 thoracic vertebrae and then the 3rd lumbar vertebrae. Okay, so the right kidney is positioned slightly lower than the left due to the presence of the liver. Now, relations of the kidney. Anterior relations. Anteriorly, the right kidney in contact with the liver. Anteriorly, the right kidney is in contact with the liver, the duodenum, and then the colon. Okay, for the left kidney, anteriorly, it is related to the stomach, the spleen, the pancreas, and then the jejunum. Okay, so we are looking at the organs that are related to the kidney. Then we are looking at in front of the kidney, anterior. So in front of the kidney, we have the right kidney is different. The left kidney is also different. So we are looking at the right kidney. For the right kidney in front, we have, it is in contact with the liver, a portion of the liver, the duodenum, and then the colon. Okay, for the left kidney, in front of the left kidney or the anterior to the left kidney, it is related to the stomach, the spleen, the pancreas, and then the jejunum. Okay, then we have the posterior relations. With the posterior relations, we are looking at the relations for both kidneys. Okay, so for both kidneys, because the kidney is not a peritoneal organ, posterior to the kidney, we are not expecting the peritoneum. We should expect muscles, blood vessels, and nerves. Okay, because the kidney is already a retroperitoneal organ. So posterior or behind the kidney, you should expect nerves, muscles, and blood vessels. Okay, so we have muscles like the diaphragm, we have the psoas major muscle, we have the quadratus lumborum muscle, we have the transversus abdominis muscle. Okay, we also have the subcostal nerves and blood vessels. We also have iliohypogastric and ilioinguinal nerves. Okay, so posterior to both kidneys, these are the possible structures that you can find there. Okay, good. Now let's look at the parts of the kidney and their functions. With this slide, we are looking at the macroscopic anatomy of the kidney. The macroscopic anatomy, or what we call the gross anatomy of the kidney. So if you are looking at the kidney in terms of if you are given a kidney and you are looking at it unaided without a microscope, the structures that you are likely to see. Okay, the structures that you are likely to see. Is that okay? Yes, so that is what this slide is displaying. So now let's concentrate on the diagram on your right. Okay, concentrate on the diagram on your right. Okay, so you see the macroscopic anatomy of the kidney. Okay, so the macroscopic anatomy of the kidney. Now we have the renal capsule. So if you look over here, you can see the renal capsule. Okay, the renal capsule is more like the outermost layer of the kidney. It is made up of fibrous tissues. Okay, so it's the tough fibrous outer covering of the kidney that provides protection and maintains the shape of the kidney. That is the renal capsule. Okay, we call it the renal capsule. Okay, the renal capsule. From the renal capsule, we have what we call the renal cortex. So this is the renal capsule, the outermost layer of the kidney. Okay, the tough fibrous outermost layer of the kidney. Okay, take note of the fact that it is not soft, it is tough. Is that okay? It is a tough fibrous outer covering of the kidney that provides protection and maintains the shape of the kidney. From the renal capsule, we have the renal cortex. Okay, the renal cortex. Okay, the renal cortex. Okay, good. The renal cortex is also the region next to the renal capsule, okay, which is also part of the outer covering of the but it comes after the renal capsule. Is that okay? And it is within this area or this region where the filtration actually begins. The reason is because we have some nephrons within this renal cortex. Is that okay? And for some nephrons, their renal corpuscles are within the renal cortex. So in anatomy, we say that blood filtration actually begins from the renal cortex. That is if we are looking at it from the capsule, using the capsule as a reference point, and you are coming inward, the filtration actually begins in the renal cortex. Then the renal medulla. The renal medulla is basically the space within the kidney. Okay, so when we say renal medulla, we are looking at the inner region of the kidney. It's like the room. The medulla is more like the room of the kidney. Okay, so within the medulla, that is where we have the renal pyramids. Okay, so if you look at this kidney, the pyramids are more like the cone-shaped structures within the medulla. Is that okay? So within the medulla, we have the renal pyramids, which are the cone-shaped structures within the renal medulla. And we have some of the nephrons within this renal pyramid. So we have the renal pyramid. Let's count them. We have 1, 2, 3, 4, 5, 6. We have, in this diagram, we have 6 renal pyramids. Okay, but in anatomy, we have about 6 to 8 pyramids in each of the kidneys. 6 to 8 renal pyramids in each of the kidneys. Okay, good. Now, at the apex or the tip of the renal pyramid, we have a structure that we call the renal papilla. Okay, so for this renal pyramid, at the apex, we have a renal papilla. If you come to this renal pyramid too, at the apex over here, we have a renal papilla. If you come here to at the apex, we have the renal papilla. The same thing applies to all the pyramids. Then from the papilla, we have a space over there or a channel, which we call the minor calyx. Okay, so each of the renal pyramids from the papilla, they all have their renal papilla, they all have their minor calyxes. Then after the minor calyx, we have another calyx we call the major calyx. So each of the renal pyramids have their individual minor calyxes and major calyxes. Then all the major calyxes of all the pyramids will join one big funnel-shaped structure that we call the renal pelvis. It's over here, the renal pelvis. Is that okay? So this renal pyramid is having a papilla, is having a minor calyx. This renal pyramid is having a papilla, is having a minor calyx. It's also having a major calyx. This renal pyramid is having a papilla, is having a minor calyx, also having a major calyx. This one too, the same thing. Then they all join, all the major calyxes will now join a funnel-shaped structure called the renal pelvis. Then the renal pelvis joins the ureter. The renal pelvis is the one that will join the ureter. Is that okay? Good. So we have the renal, from the renal, we have the renal papilla, then we have the minor calyx and then the major calyx. The calyxes are more like structures that collect urine from the renal papilla. We have the minor calyxes, okay, which merge to form the major calyxes. Then the major calyxes drains into the renal pelvis. Then we have the renal pelvis. The renal pelvis, like I said in my explanation, is a funnel-shaped cavity that collects urine from the calyxes and channels it into the ureter. Okay, then this region, the region where you see the blood vessels, the renal artery, the renal nerves, okay, the renal veins entering the kidney, this region over here, this region that I'm going around, we call this region the renal ileum. So you see it has been labeled over here. So the entry and exit sites on the medial side of the kidney. This is the medial side, the middle side of the kidney, where the blood vessels, the lymphatics, the nerves, and then even the ureter or the renal pelvis enters. That area or region is what we refer to as the renal ileum. So if you are asked to talk about the macroscopic anatomy of the kidney, this is the description you are going to give. The renal capsule, the renal cortex, the renal medulla, the renal pyramids, the renal papilla, the calyxes, the renal pelvis, and then the renal ileum. Okay, so this is basically the gross anatomy of the kidney, the gross anatomy of the kidney. So once again, we have about 8 parts of the gross anatomy: the renal capsule, the renal cortex, the renal medulla, the renal pyramids, the renal papilla, the calyces, the minor and the major calyx, the renal pelvis, and then the renal ileum. Okay, the gross anatomy. So if you look over here, you see the minor calyx is over here, the major calyx is over here, then all the major calyxes will join the renal pelvis. Is that okay? Basically, this is the gross anatomy of the kidney. Now let's move on to the next slide. This is also showing the gross anatomy. When we say gross anatomy, it's just the same as the internal anatomy of the kidney. So this slide is also showing the internal anatomy of the kidney. So like I said, this region where the blood vessels, the renal artery, the renal vein, the renal nerves are entering the kidney. This is the medial aspect. It's more like the entry and exit point of the kidney. We call this region the renal ileum. Is that okay? Then we have, this is the cortex. Is that okay? The cortex. Then the outermost cavern made up of fibrous tissue is referred to as the renal capsule. Okay, then this is the ureter. This is the renal pelvis. So we have the minor calyx, the major calyx, the renal pyramid, the renal papilla, the tip of the renal pyramids. It's also shown over here. Is that okay? So basically, this is the internal anatomy of the kidney. Now, let's go on to the main deal, the microscopic anatomy of the kidney. The microscopic anatomy of the kidney. So now, we've looked at the part of the anatomy that you can see unaided. But on this slide, we are going to look at the part of the anatomy that you cannot see with your naked eye. Okay, you need a microscope. And that is what we call the microscopic anatomy of the kidney. In fact, remember that anytime you hear microscopic anatomy of the kidney, we are referring to anatomy of the nephron. So anytime you hear microscopic anatomy of the kidney, we are talking about the nephrons. Okay? The nephrons. Is that okay? So anytime you hear microscopic anatomy of the kidney, is that okay? We are talking about the nephrons. Okay, good. So always remember, the moment you hear microscopic anatomy of the kidney, all they are talking about is the nephrons. That's the microscopic anatomy of the kidney. And this is how the nephron look like. The nephron looks like this. Okay, and so we are coming to talk about this structure, the nephrons. Now, the nephrons are basically the functional unit of the kidney. Okay, the functional unit. Okay, we mentioned that the kidney is more like the primary organ of the urinary system. Okay, so most of the major functions or the functions of the urinary system is performed by the kidneys. Okay, and we have the nephron within the kidney. That ability or that functions that are performed by the kidneys, based on which we can say that the kidney is the primary organ of the urinary system, is because of the nephrons that are within the kidney. And that is why we are saying that the nephrons are actually the functional unit of the kidney, and that the nephrons are the ones that are responsible for filtration, reabsorption, secretion, and then excretion. In fact, in each kidney, we have about 1 million nephrons in each kidney. That should tell you that in both kidneys, you should estimate about 2 million nephrons in both kidneys. Now, these are the parts of the nephrons. We have the Bowman's capsule, we have the glomerulus, we have the proximal convoluted tubule, we have the lobe of Henle, we have the distal convoluted tubule, and then we have the collecting duct. Okay, good. So let's go back to the diagram. Okay, so these are the parts of the nephrons and then their function. So we started by saying that we have the glomerular capsule. This structure that looks like a funnel shape is what we call the glomerular capsule. Then we have this structure within looking like network of capillaries. We call it glomerulus. It looks more like cluster of capillaries, cluster of tiny blood vessels. we call it the glomerulus. And the glomerulus is within the glomerular capsule. The glomerular capsule is also called the Bowman's capsule. Okay, the glomerular capsule is also called the Bowman's capsule. Okay, good. So this is the glomerulus. This is the glomerular capsule, which we also call the Bowman's capsule. Then we have the proximal convoluted tubule. So from the Bowman's capsule, the tubule continues. We have the proximal convoluted tubule. And we call this proximal convoluted tubule because when we are using this Bowman's capsule, we use this Bowman's capsule as a reference point. We call this proximal convoluted tubule because if you are using the Bowman's capsule as a reference point, this part of the tubule is closer to the glomerular capsule or the Bowman's capsule. And that is why we call it proximal convoluted tubule. From proximal convoluted tubule, okay, it descends. Okay, so from proximal convoluted tubule, we have the loop of Henle. This from here rising upwards to this point. So from where the arrow begins, descending downwards, rising upward to this point is what we call the loop of Henle. So the loop of Henle in anatomy has two parts. This part coming down is the descending limb. So you can see that labeled over here, descending limb of the loop of Henle. Then the part going up, we call it the ascending limb of the loop of Henle. So we have the descending limb of the loop of Henle, and then we have the ascending limb of the loop of Henle. The descending limb of the loop of Henle, and then we have the ascending limb of the loop of Henle. Is that okay? So we have the descending limb of the loop of Henle, and then we have the ascending limb of the loop of Henle. Then from the ascending limb of the loop of Henle, we have the distal convoluted tubule. We have the distal convoluted tubule. From the distal convoluted tubule, we have the collecting duct. The collecting duct. Is that okay? The collecting duct. If you look on your left, you can see that the cortex is over here, and then the medulla is over here. So you can see that if you look at this nephron, okay, we have some of the nephron within the cortex and a portion of the nephron also within the medulla. And we will talk about that later. It's important that you know that because we are going to talk about that later on as we move on. Now, let's look at the part of the nephron again into detail. So we have the Bowman's capsule. And another thing I didn't mention is that this is the Bowman's capsule or the glomerular capsule. This is the glomerulus. Now, this region where we have the glomerular capsule or the Bowman's capsule and the glomerulus, this region is what we refer to as the renal corpuscle. Renal corpuscle. So renal corpuscle is like the region where we have the glomerulus within the glomerular capsule or the Bowman's capsule. This region is what we call the renal corpuscle. Now, you can see that there is an arteriole bringing blood into the glomerulus. This arteriole bringing blood into the glomerulus to form the capillary network of blood vessels called the glomerulus. This artery bringing it inside is called the afferent arteriole. And we have another artery taking the blood away or rising away from the glomerulus. That artery is called the efferent arteriole. Efferent arteriole. So we have the efferent arteriole and then we have the afferent. The afferent is what brings the blood inside the glomerulus. It is bringing it inward. And then the efferent is taking it outside. Is that okay? The efferent is taking it outside. The afferent is bringing it inside. Okay, so we have the afferent bringing it inside. The efferent taking it outside. Okay. Good. Now let's look at the parts of the nephron into detail. So we have the Bowman's capsule. The Bowman's capsule is more like the cap-like sac that surrounds the glomerulus. This sac. See, this is the glomerulus. So that cap-like sac, more like a funnel-shaped structure that surrounds the glomerulus. That is what we call the Bowman's capsule or the glomerular capsule. Okay. In fact, it serves as a site for filtration of the blood. Okay, so the blood enters the Bowman's capsule through the afferent arteriole and is filtered through the glomerular capillary membrane, which is the glomerulus, and that forms a filtrate free of cells and large proteins. Then we have the glomerulus. Okay, we have the glomerulus itself. Glomerulus is like a cluster of fenestrated capillaries within the Bowman's capsule. This is what we call the glomerulus. This region is what we call the glomerulus. Look at it very well. This region is what we call the glomerulus. You see, it is within the glomerular capsule or the Bowman's capsule. Okay, so it's made up of a cluster of fenestrated capillaries within the Bowman's capsule. And this is where ultrafiltration or filtration actually take place. Filtration of water, ions, glucose, amino acids, and small molecules pass through into the Bowman space. Whilst protein, so what we are saying is that in it filtering the blood, certain substances are able to pass through it, whilst other substances are not allowed to pass through because of their size or their charges. Okay, and in fact, you see it composite more like something that has been folded like a ball, but in actual sense, it's in 3 layers. Okay, and the layers, we have fenestrated endothelium or endothelium of the capillaries. We have the glomerular basement membrane or the basement membrane, and then we have the podocyte foot processes, or what we call the epithelial layer. Is that okay? So in actual sense, in effect, you see it to be something composite like a ball, like something unit, like a unit component of a structure. But then in actual sense, it's in layers of 3. And because of that, we can even call it tri-layered filtration barrier or tri-layered filtration membrane. That is another name, tri-layered filtration membrane. But one important thing you have to remember is that this glomerulus is what we also refer to as the glomerular filtration membrane. This is what forms the glomerular filtration membrane. And I'm saying that it's in 3 layers. We have the endothelium of the capillaries, the glomerular basement membrane, and then the podocyte foot process. Okay, we will come to that and look at that into detail later on. Now we have the proximal convoluted tubule. The next structure is the proximal convoluted tubule. This is the proximal convoluted tubule. It is proximal because we are using the glomerular capsule as the reference point, and because it is closer, that's why we say it is proximal. Okay, so the proximal convoluted tubule, over there, a lot of reabsorption take place. Okay, it has that ability to, or that role of reabsorbing 65 to 70% of the entire filtrate. So let's say the 100% filtrate that goes into the Bowman's capsule, when they get to the proximal convoluted tubule, about 65 to 70% of them are reabsorbed back into the blood. Okay, apart from that, substances like glucose, amino acids, sodium, chloride, bicarbonate, water. Is that okay? Good. These substances sometimes can be reabsorbed back at the level of the proximal convoluted tubule. Okay. In the proximal convoluted tubule too, some amount of secretion can also take place. Secretion of hydrogen ions. Hydrogen ions can be secreted into the proximal convoluted tubule, and some drugs or toxins can also be secreted back into the blood via the proximal convoluted tubule. Then we have the loop of Henle. Okay, the loop of Henle. The loop of Henle, like I said earlier, is made up of 2 parts. So this is the loop of Henle from here rising up. So the part descending is called the descending limb of the loop of Henle. Then the part rising is referred to as the ascending limb of the loop of Henle. Is that okay? Good. Another key thing that you should remember is that in the proximal convoluted tubule, I mentioned that a lot of reabsorption can take place. But apart from that, secretion of certain drugs from the peritubular capillaries can occur, whereby drugs, toxins, excess hydrogen ions can be secreted back into the proximal convoluted tubule. In fact, one other unique thing is that in the proximal convoluted tubule, about 60 to 70% of water that forms part of the filtrate are reabsorbed back into the peritubular capillaries. And this is essential because if that do not happen, you can dehydrate. And because you see, water is a freely moving particle which can easily pass through the glomerular filtration membrane. So let's assume that all the water you drink finally makes their way into the proximal convoluted tubule. Think about that. And then they end up coming out to form part of the urine. You end up urinating all the water in your blood away and get dehydrated, because at the glomerulus over here, okay, even though blood cells are not permitted to pass through the filtrate, but plasma is being filtered. The plasma component of the blood is being filtered. Okay, and the plasma, with the exception of some large proteins, all of them are being filtered. Is that okay? Good. So we have the loop of Henle. At the loop of Henle, reabsorption also take place at the loop of Henle. So reabsorption specifically of water, is that okay, take place over there. Okay, and then at the ascending limb to reabsorption of sodium, reabsorption of potassium, chlorine, okay, can also occur at the ascending limb of the loop of Henle. Then we have the distal convoluted tubule. The distal convoluted tubule also helps to fine-tune sodium, potassium, and calcium by regulation under the regulation of aldosterone release, which we will look at extensively as we move on. Okay, good. So at the distal convoluted tubule, reabsorption of sodium, potassium, calcium ions. At the distal convoluted tubules too, there can be secretion of excess ammonia, drugs, and toxins can also take place over there. Then we have the collecting duct. Now, even in the collecting duct, so once the filtrate reaches the collecting duct, then it's almost getting into excretion. So at the collecting duct, there can be reabsorption of water sometimes in situations where your body seriously needs them. There can be reabsorption of water. There can also be reabsorption of sodium ions. Is that okay? Good. Then again, there can also be secretion at the collecting tubule or collecting duct. There can be secretion or reabsorption of hydrogen ions or bicarbonate ions depending on the body, and it's because of this collecting duct, the process that goes on there. That is why we say the kidney plays a role in regulating acid-base balance. You see, we have electrolyte balance and we have acid-base balance. Is that okay? Because you see, at the collecting duct, is that okay? There can either be secretion or reabsorption of what? Hydrogen ions and bicarbonate ions, and it is these two ions, their concentration is what determines your acid-base balance in your body. Okay, then the last aspect of the nephron is you see some tiny blood vessels around the tubules. We call them peritubular capillaries. Peritubular capillaries. Okay, good. These capillaries are the capillaries that return. In fact, they usually return from the efferent arteriole. Is that okay? Good. And they come and play a role in the reabsorption of water, the solutes, and then the ions. Is that okay? For the reabsorption process. Okay, so basically these are the primary organs, the organs of the structures within the nephron. Structures within the nephron. So anytime you hear microscopic anatomy of the kidney, remember that we are talking about the nephrons. Okay, anytime you hear microscopic anatomy of the kidney, we are talking about the nephrons. Okay, yes. So in summary, the nephron has about 6 parts. We have the Bowman's capsule and then the glomerulus. These two together forms the renal corpuscle. Okay, and this glomerulus, even though you see it like this, in effect, is actually in 3 layers. So the 3 layers, we have the fenestrated endothelium of the capillaries or endothelium of the capillaries. We have the glomerular basement membrane. We have the podocyte foot process. Okay, we have the proximal convoluted tubule PCT. Okay, which helps to reabsorb about 65 to 70% of the entire filtrate. Okay, and the reabsorption includes reabsorbing substances like glucose, amino acids, sodium chloride, bicarbonate, water, and secrete hydrogen ions and drugs. Then we have the lobe of Henle. Okay, the lobe of Henle, we have the descending limb of the lobe of Henle, the ascending limb of the lobe of Henle. Then we have the distal convoluted tubule, DCT. Okay, which helps to fine-tune sodium, potassium, calcium, and regulate under the regulation of aldosterone and then parathyroid hormone. Okay, and plays a role in maintaining your acid-base balance. And the last one is the collecting duct, which basically collects the urine from the nephrons. Okay, and under the regulation of antidiuretic hormone, it can play a role in water reabsorption. Okay, and then it also helps to maintain your acid-base balance, because over there secretion of what we call secretion of hydrogen ions and bicarbonate ions can occur depending on your body's acidity level or your body's pH level. Okay, good. So basically that is the nephrons. That is the nephrons. Now we have two main types of nephrons. The two main types of nephrons, we have the cortical nephrons, and then we have the juxtamedullary nephrons. Okay, we have the cortical nephrons, and then we have the juxtamedullary nephrons. Okay, when we say cortical nephrons are nephrons that a greater portion of their loops, okay, or a greater portion of their tubules, okay, or a greater portion of it is within the cortex of the kidney. Then we have the juxtamedullary nephrons. These are nephrons that are basically have a greater portion of their tubules or their loops within the medulla. That is the juxtamedullary nephrons. So we have two main types of nephrons, cortical nephrons and juxtamedullary nephrons. Okay, in fact, if you are looking at all the nephrons in the kidney, about 85% of the nephrons are cortical nephrons, and about 15% of nephrons are juxtamedullary nephrons. That tells you that cortical nephrons actually dominate. So, and that is the reason why when we were looking at the macroscopic structure, we gave the renal cortex that recognition, that because of this reason, because you see most of the nephrons are even within the cortex. And so if you are talking about the renal cortex, you should be able to give it that recognition or respect, because most of the nephrons are within the cortex. So because of that, the filtration of the blood actually begins from the renal cortex. Okay, it constitutes about 85% of all the nephrons in the kidneys. And then the juxtamedullary nephrons constitute about 15% of all the nephrons in the kidneys. Okay, so basically that is it. Is that okay? Basically that is it. Good. Now let's look at the nephron in a different perspective, to look at some other key features or key characteristics of the nephron. Now this is the nephron. Okay, this is the nephrons. This diagram is showing the nephron. So we are looking at the two main types of nephron. So with the two main types of nephron, we have the cortical nephron. With the cortical nephron have a greater portion of their tubules or loops within the cortex. So if you look at this nephron, you can see greater portion of their tubules are within the cortex. So you can see a greater portion of their tubules. If you look at this nephron, greater portion of their tubules are within the cortex. And if you look at this one, you can see that greater portion of their tubules are within the medulla, because from here to this side is the medulla. It's shown on our left. From this side to this side is the medulla. So if you look at it critically, you realize that a greater portion of this nephron is within the medulla. But if you come over here, you could see a greater portion of the tubules of this very nephron is within the cortex. If you come to this diagram to the same, from here to here is the cortex. From this side to this side is the medulla. So you can confirm from this diagram that this nephron is actually a juxtamedullary nephron. Okay, juxtamedullary. You get it? Juxtamedullary nephron. Is that okay? Good. And this nephron is also a cortical nephron, because the reason it's a cortical is because, see, if you look over here, you see they've even written cortical glomerulus. Okay, if you look over here, they've even written juxtamedullary glomerulus. So this one is a cortical nephron, and this one is a juxtamedullary nephron. Okay, so because it's a juxtamedullary nephron, its glomerulus is called juxtamedullary glomerulus. And here, because it's a cortical nephron, its glomerulus is also called cortical glomerulus. Is that okay? Good. So basically that is it. Okay, now the key thing I want you to remember is that, you see, when this is the glomerulus, this is the Bowman's capsule. The glomerulus is within the Bowman's capsule over here, and this is the proximal convoluted tubule winding around itself, coming to this side. Then when it is descending, this is the loop of Henle. So this is more like the descending limb of the loop of Henle. Then it rises, the ascending limb of the loop of Henle. Now remember that the distal convoluted tubule, so when the ascending limb of the loop of Henle rises and starts the distal connects to the distal convoluted tubule, the distal convoluted tubule always forms a contact with the afferent arteriole. And that is one of the ways that you can confirm or tell that you can confirm or actually tell that this is an afferent arteriole and this is a distal convoluted tubule. Okay, so the distal convoluted tubule always forms a contact, and that region or that area where it forms the contact, we call that region the juxtaglomerular apparatus. Okay, juxtaglomerular apparatus. Now let's look at it from this diagram. If you come and look over here, you can see this is the glomerulus and this is the Bowman's capsule or the glomerular capsule. This tubule over here is the proximal convoluted tubule. You see it has even been labeled proximal convoluted tubule. Then when it starts coming down, from here coming down is the loop of Henle, descending limb of the loop of Henle. Here is the ascending limb. Then when it gets here, here is the distal convoluted tubule. You can see that the distal convoluted tubule forms an attachment over here. Look into the cursor. You can see there is a junction over here. This is what we call the juxtaglomerular apparatus, where the distal convoluted tubule actually forms contact with the proximal. So always in anatomy diagram, very well represented anatomy diagrams, you should see the distal convoluted tubule actually forming an attachment with the afferent arteriole, and this would help you to know that it's actually an afferent arteriole. It will also help you to know, and it will also help you to know that attachment will also help you to know that this one is also a distal convoluted tubule, because of the fact that this is the one forming an attachment. So if you look at this diagram carefully, you can now confirm that, okay, then this arteriole over here is the afferent arteriole, and this arteriole over here is the efferent arteriole. Is that okay? The same thing. So if you come and look over here, you see this has been labeled the afferent arteriole, and this one has been labeled the efferent arteriole. Is that okay? So basically, that is one other key thing about the afferent arteriole. Okay, and remember that within this afferent arteriole, okay, we have some cells in the afferent arteriole. We call it the juxta medullary cells. Okay, and then within this distal convoluted tubule, we have some cells inside. We call it the macula densa cells, and it's important that you remember, because we will come to those cells and then their relevance. Okay, and we'll talk about it. Good. Now let's look at the glomerular filtration membrane. The glomerular filtration membrane. The glomerular filtration membrane. In fact, the glomerular filtration membrane, or in some literatures we call it the glomerular capillary membrane, is the structure in the kidney that facilitates the filtration of blood to form the urine. In fact, this structure is actually part of the glomerulus, which is a network of capillaries located in the renal corpuscle of the nephron. Okay, so in fact, the glomerulus, this is what actually forms this glomerulus. It has been labeled over here, glomerulus. This is what actually forms the glomerular filtration membrane. So anytime you hear glomerular filtration membrane, your focus should be over here, the glomerulus, which is within the Bowman's capsule. And I told you that even though you see it composite like network of capillaries, but in essence it's actually in 3 layers, and that is what we are coming to talk about on this slide, the glomerular filtration membrane. So when you hear glomerular filtration membrane, we are talking about the glomerulus. Is that okay? It is the structure in the kidney, specifically the nephrons, that facilitate the filtration of blood to form the urine. Okay, it is part of the glomerulus, a network of capillaries located in the renal corpuscle of the nephrons. Now, we also call it tri-layered filtration barrier. And the reason why we call it a tri-layered filtration barrier is because it is made up of 3 layers. And the layers include 1, the endothelium of the capillaries, 2, the glomerular basement membrane or the basement membrane, and then 3, the epithelial cells or the podocytes. Okay. Now, let's look at it from these diagrams. So we have the endothelium of the capillaries over here. This is the first layer, or this is the innermost layer. Okay, so I explained the other time that if we are using the blood or the capillary lumen or the glomerulus as a reference point, or the capillary lumen as a reference point, then this is the innermost layer. But if you are speaking and you are using the filtrate as a reference point, then this epithelial cells or the podocytes will be the innermost layer. But in this situation, we are using the blood, okay, as the reference point. So this happens to be the the first layer or the innermost layer. So the innermost layer is the endothelium of the capillaries. Endothelium of the capillaries. From the innermost layer, the middle layer is the glomerular basement membrane, and then the last layer is the last layer happens to be the epithelia. Sorry, the last layer happens to be the epithelial cells or the podocytes. Now, in between the first layer, look at it very well. In between the first layer, okay, or even before the first layer, we have a layer of tiny protein structure in front of the endothelium of the capillaries. We call it the glycocalyx. It's made up of proteins. Then we have the glomerular basement membrane over here. Glomerular basement membrane. Glomerular basement membrane. From the glomerular basement membrane, we have the epithelial cells or the podocytes. Epithelial cells or the podocytes. Epithelial cells or the podocytes. Okay, in between the endothelium of the capillaries, which is the innermost layer, we have spaces over there. You can see we have space over here, 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 we have another space over here. Okay, so in between the endothelium of the capillaries, we have spaces in between. Is that okay? Good. And those spaces, we call those spaces fenestrations or pores. Fenestration or pores. Then we have the glomerular basement membrane. Okay, or the basement membrane. This is the middle layer, and it is composed of a gel-like matrix of proteins. In fact, this layer is what serves as the major barrier, okay, for selective filtration. Okay, then from there we go to the outermost layer, okay, which we refer to as the podocytes or epithelial podocytes or epithelial cells. So these are the podocytes. So this podocyte, 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 podocyte. This is also a podocyte. This is another podocyte. This is another podocyte. And it is aligned by what we call the slit diaphragm. Is that okay? Then we have spaces in between them. We call the filtration slits. Okay, so we have spaces in between the epithelial cells or the epithelial podocytes that we call the filtration slits. So this is what we call the glomerular filtration membrane. Anytime you hear glomerular filtration membrane, it's the same as the glomerular filtration barrier or glomerular capillary membrane. Or you can also call it the tri-layered filtration barrier. Tri-layered filtration barrier. Okay, tri-layered. Why is it called tri-layered filtration barrier? Because it is made up of 3 layers. Okay, the first and innermost layer, using the blood as the reference point, is the endothelium of the capillaries. The second layer is the glomerular basement membrane. And then the third layer is the epithelial cells or the epithelial podocytes. Or you can simply call it podocytes. It's the outermost layer if you are using the blood as the reference point. And these podocytes have foot-like processes called the pedicels, which interlock to form filtration slits. Is that okay? They interlock to form the filtration slits, which further regulates the filtration. Okay, so this is what we call the glomerular filtration membrane. This is a glomerular filtration membrane. This membrane is very important, especially to help you appreciate certain disorders that affect especially the kidneys or the nephrons. It's very important for you to understand this glomerular filtration membrane. So in future, if you are learning any medical condition, and then we mentioned of the fact that this condition damages the glomerular filtration membrane, this is the membrane that we are making reference, we are talking about. Is that okay? And this is formed by the glomerulus, which is within the renal corpuscles. Okay, so basically this is it for the glomerular filtration membrane. Now, let's look at glomerular filtration rate. Glomerular filtration rate, GFR, glomerular filtration rate. When we say glomerular filtration rate, we are looking at the amount of filtrate that is formed by each kidney per minute. The amount of filtrate that is formed by each kidney per minute. That is what we call the glomerular filtration rate. Okay, so when we say glomerular filtration rate, we are looking at the amount of filtrate that is formed by each kidney per minute. Okay, so always remember, the amount of filtrate that is formed by each kidney per minute is what we refer to as the glomerular filtration rate. Okay, now if we are looking at it in a range for a healthy adult, we expect that the glomerular filtration rate should be between 90 to 120 mL per minute for a healthy adult. Okay, in a range, it should be between 90 to 120 mL per minute. Per conversion is the same as 1.5 to 2.0 mL per second. 1.5 to 2.0 mL per second per conversion. Okay, but then if you want it in a unit or a whole, okay, or as in an average, okay, the normal glomerular filtration rate for a healthy adult is 125 mL per minute. 125 mL per minute. Now, the normal kidney forms approximately 1 to 2 liters of urine per day in a healthy adult, depending on fluid intake, the individual's activity levels, and then the overall health of the individual. Okay, so for a normal kidney, it forms approximately 1 to 2 liters of urine each day, especially in healthy adults, depending on fluid intake, the activity levels of the person, and then the overall health of the person. Okay, now remember, we have something we call creatinine clearance. Creatinine clearance is actually an excellent measure of renal function. Okay, because as renal function declines, creatinine clearance also decreases. Okay, so when we say creatinine, it's simply a waste product or an end product from skeletal muscle metabolism. Okay, and it's usually filtered at the glomerulus. Okay, now let's look at some 8 substances. Okay, some 8 substances that can be excreted by the kidneys. Okay, good. Let's look at some 8 substances that can be excreted by the kidneys. Okay, yes, so look on the slide. The 8 substances: we have urea, we have creatinine, we have uric acids, we have ions, we have electrolytes, we have hydrogen ions, okay, we have ammonia, we have drugs, and we have toxins, and then we have excess water. So all these substances, they can be excreted by the kidney. This is usually a waste. Okay, this is a waste. Urea is a waste product. Creatinine is also a waste product. Uric acid is also a waste product from purine metabolism. Ions excess can be a waste. For ions and electrolytes, you can't say it's a waste. Okay, but if it is in excess, then it can be a waste. Hydrogen ions, if it's also in excess, it can be a waste. Ammonia is also a byproduct from nitrogen metabolism. Okay, drugs, toxins, excess water. Okay, and this is the reason why we are saying that this creatinine is actually the best if you want to actually confirm that the kidney is really working well. You have a patient and you want to confirm that the person's kidney is really functioning well. The best to focus on is the creatinine, because this creatinine is not dependent on any factor. It's dependent on limited factors. For example, whether you eat is just the end product of muscle metabolism. So because you see, whether you eat, whether you don't eat, whether you exercise, you don't exercise, whether you choose to sleep the rest of your life, still creatinine will be produced. Because even if you are sleeping your whole life, your respiratory system, your respiratory muscles are working. Is that okay? Yes, your respiratory muscles, intercostal muscles, they work. So still, creatinine will be produced because creatinine is the end product of skeletal muscle metabolism. You get? So whatever you choose to do, still creatinine will be produced. Is that okay? Whatever you choose to do, whatever you choose to do, still creatinine will be produced. So therefore, if you really want to determine that a patient is having a problem, okay, a particular patient is having a problem with renal problem, the best measure to focus on is creatinine clearance. Okay, creatinine clearance. Because, for example, if you choose to say that you are opting for urea, it's also good anyway. Okay, because if urea accumulates, it can also be that the kidney is not functioning well. But another thing is that urea can be influenced by a lot of factors. You see, it's a byproduct from protein metabolism. What if the person doesn't really consume proteins? Is that okay? So it can influence it to become low, not necessarily coming from a problem with the kidney. Okay, good. Good. Another thing is uric acid. Uric acid is a byproduct from pouring metal. What if the person is not consuming foods that, ends up in pouring? Is that okay? Good. So that is basically, so the best is creatinine clearance. Just that for creatinine can also depend on factors like age. Okay, creatinine can also depend on factors like age, sex, and muscle mass. So for example, if you take the creatinine for a child of, let's say, 1 year, you can compare it to the creatinine levels of an adult because of the differences in their muscle mass. Is that okay? Because of their differences in their muscle mass. Okay, so creatinine, even though it's very important, so this is what I'm talking about. So look at it and think about it properly. Which is important? BUN and then BUE, which is important? Blood urea estimation, and then creatinine. Which of them is very important? Is that okay? Which of them is very important? The important one is creatinine. The reason is because creatinine measures the level of creatinine clearance measures the level of creatinine, which is a byproduct of muscle metabolism. Is that okay? So creatinine is more specific marker of kidney function compared to BUN, like I was explaining. The reason is because it reflects the kidney's ability to filter waste without being significantly affected by factors like diet or hydration. Is that okay? Good. So whether you drink water, whether you don't drink water, still creatinine will be produced. Whether you choose to eat, whether you choose not to eat, it's not influenced by factors like diet, unlike urea and then uric acid and other. So for men, okay, creatinine levels, normal creatinine levels should be between 0.7 to 1.3 milligram per deciliter. Okay, and per conversion, this is it. For women, it's between 0.6 to 1.1 milligram per deciliter. So, because men relatively tend to have more muscle mass. Is that okay? Yes, so we expect that definitely the creatinine levels would be higher in men as compared to women. For children, it is relatively very low, 0.3 to 0.7 milligram per deciliter. Okay, now if creatinine and urea accumulate in the blood, it indicates impaired kidney function. Is that okay? And can lead to a toxic condition called uremia. Okay, in the long run, this condition can damage organs and lead to life-threatening complications. Okay, so basically, this is what I was talking about. Good. Now, let's look at filtration pressure and glomerular filtration rate in the kidneys. Filtration pressure and glomerular filtration rate in the kidneys. Okay, filtration pressure and glomerular filtration rate in the kidneys. Now, we have three main pressures. When we say glomerular filtration rate, we are looking at the amount of filtrates formed by the kidneys per minute. Okay, and that pressure actually depends on three pressures. Is that okay? Now we are going to look at these three pressures and how we can consider these pressures to arrive at the glomerular filtration rate. In fact, when we say glomerular filtration rate, if you want to calculate glomerular filtration rate, the formula we use is glomerular filtration rate is equal to net filtration pressure times the filtration coefficient, KF. Okay, net filtration pressure, which is the NFP, multiplied by the filtration coefficient. Okay, good. And usually, this KF is usually constant. It's usually 12.5 mils per minute per millimeters of mercury. Do you get it? 12.5 mils per minute per millimeters of mercury. That's the filtration pressure. 12.5 mils per minute per millimeters of mercury. Okay, now let's look at the filtration pressures. Now we have three pressures that contribute or help us to arrive at the glomerular filtration rate. The first pressure, we call it the glomerular blood hydrostatic pressure. The glomerular blood hydrostatic pressure or blood hydrostatic pressure in the glomerulus. Glomerular blood hydrostatic pressure or blood hydrostatic pressure in the glomerulus. Okay, glomerular blood hydrostatic pressure or blood hydrostatic pressure in the glomerulus. In fact, this is the main outward pressure. This pressure is actually an outward pressure. Okay, good. So the blood hydrostatic pressure in the glomerular capillaries, this pressure is usually about 60 millimeters of mercury. In some literatures, they will say it's about 55 millimeters of mercury, which is much higher than the pressure in other capillaries. Okay, and this pressure is very high because of the nature of the afferent arteriole. The afferent arteriole is very wide, and that is what makes this pressure very high. Okay, good. Then we have two other pressures. We have the capsular hydrostatic pressure, which is usually between 15 millimeters of mercury or 18 millimeters of mercury. Okay, good. This is the pressure from fluid in the capsule pushing the fluid back into the glomerulus. Okay, we call that the capsular hydrostatic pressure. Then we have the colloid osmotic pressure or oncotic pressure within the glomerulus, and that pressure is about 32 millimeters of mercury. That pressure is about 32 millimeters of mercury. Okay, now for anatomy, okay, the key thing is not for you to know the values, but for you to be able to tell where do we have the glomerular blood hydrostatic pressure. So the glomerular blood hydrostatic pressure is within the glomerulus. Okay, it's coming from the glomerulus, it's coming from the efferent arteriole towards the glomerulus. Where do we have the capsular hydrostatic pressure? Within the Bowman's capsule. Okay, which is bringing it, that pressure is pushing the fluid within the Bowman's capsule back into the glomerulus. Okay, then we have the osmotic or oncotic pressure. Okay, and this pressure is within the glomerulus. This pressure pulls water back into the blood due to the proteins in the plasma of the blood. Okay, and it's also an opposing pressure. It opposes the glomerular blood hydrostatic pressure. Okay, so now if you want to calculate the glomerular filtration rate, okay, what you do is that, if you want to calculate the glomerular filtration rate, you first of all have to get the net filtration pressure. How do you get the net filtration pressure? Before you get the net filtration pressure, you have to add the two opposing pressures or the two inward pressures. You add the two opposing pressures or the two inward pressures. The two opposing pressures is either you add 18 plus 32 or you add 15 plus 30. Either you add 18 plus 32 or you add 15 plus 30. Is that okay? Good. They are the two opposing pressures. Okay, good. So the two opposing pressures, you add the capsular hydrostatic pressure to the colloid osmotic pressure or the oncotic pressure. You add these two. Okay, then the answer that you get, you subtract it from the glomerular blood hydrostatic pressure. You subtract it from, then you get the net filtration pressure. Now, when you get the net filtration pressure, usually if you are using a healthy adult, if you add 18 to 32, you are going to get 50. And if you subtract 50 from 60 millimeters of mercury, at the end of the day, you should get a net filtration pressure of 10 millimeters of mercury. And a net filtration pressure of 10 millimeters of mercury is normal for a healthy adult. It's very normal for a healthy adult. Now, another thing is that let's do with the 55. If you are using glomerular blood hydrostatic pressure of 55, okay, and then the capsular hydrostatic pressure is 15. So 15 plus 30 will give us 45. If you subtract 45 from 55 too, you realize that the net filtration pressure, you still get 10. Is that okay? Good. So to calculate for the glomerular filtration rate, is 10 millimeters of mercury times the coefficient. So 10 millimeters of mercury times the 12.5 mils per minute per millimeters of mercury. So the millimeters of mercury will cancel the millimeters of mercury for the net filtration pressure. Then the final unit is going to be mils per minute. So in doing calculation for glomerular filtration rate, the final unit should be mils per minute. The final unit should be mils per minute. The final unit should be mils per minute. Always remember that the final unit should be mils per minute when you are doing calculation for glomerular filtration rate. Okay, so in summary, we have three pressures that plays a role. In fact, the greatest of all the pressure is 60 millimeters of mercury, which is the glomerular blood hydrostatic pressure. Then the second largest pressure is the colloid osmotic pressure, which is about 32 millimeters of mercury. Then the next is the capsular hydrostatic pressure, which is about 18 millimeters of mercury, or 15 millimeters of mercury. Here can be 30 millimeters of mercury. Okay, and here can also be 55 millimeters of mercury. If you are using either of them, at the end of the day, you end up with a normal glomerular filtration rate of about 10, a net filtration pressure of about 10 millimeters of mercury. Then you multiply that by the coefficient of filtration, which is 12.5 mils per minute. Okay, so the glomerular filtration rate is the total volume of filtrate formed per minute by both kidneys. The average is 125 mils per minute. That is what I was explaining, 125 mils per minute, especially in males, and 105 mils per minute in females, totaling 150 to 180 liters per day. Okay, 150 to 180 liters per day. So in a day, this amount of filtrate is formed by the kidneys in a day. But because of reabsorption, you shouldn't expect the person to urinate all of this away in a day. Okay, so basically that is it for the filtration pressure and then the glomerular filtration rate in the kidney. So this is a diagram. So you can see the greatest pressure. You can see the yellow showing the hydrostatic pressure in the glomerulus. You see, that is the largest. If you look at the arrow, and this is an outward pressure. And you see the second largest is the oncotic pressure, which is the 32 millimeters of mercury. That is the colloid osmotic pressure in the glomerulus. That is the second largest, which is 32. In this diagram is 30. And the largest one in this diagram is 55. Okay, these are all for healthy adults. Okay, then we have the hydrostatic pressure in the capsular hydrostatic pressure. That is the 18 millimeters of mercury. So if you want to calculate the net filtration pressure, okay, you add the colloid osmotic pressure to the capsular hydrostatic pressure, then you subtract it from the blood hydrostatic pressure in the glomerulus, or the glomerular blood hydrostatic pressure. And usually for a healthy person, you should get 10. So the net filtration pressure will be 10. So if you do the same calculation for this side, so you get the same thing. Net filtration pressure will be 10. Then at the end of the day, if you want to calculate the glomerular filtration rate, is the net filtration pressure multiplied by the coefficient of filtration, which is 12.5. Then you get 125 mils per minute. 125 mils per minute. Now let's look at renal autoregulation. Renal autoregulation. Okay, renal autoregulation. Okay, renal autoregulation. Renal autoregulation. Okay, now renal autoregulation is more like a mechanism that helps to keep the glomerular filtration rate in a stable state despite changes in an individual's blood pressure. Okay, so anytime you hear, because see, if your blood pressure is too high, at the end of the day, your glomerular filtration rate would also be too high. And then filtrate will form so fast, fluid will move too fast. And when fluid is moving too fast because of high glomerular filtration rate, there is going to be poor reabsorption, and that would contribute to dehydration. Okay, and when fluid is moving too low, okay, what will happen is that there is going to be very low filtrate forming, and that will mean that a lot of toxins can accumulate within the blood. Is that okay? Good. So, there should be some checks, and that process is what we call the renal autoregulation, whereby the kidney is able to, sort of regulate the glomerular filtration rate all the time. Okay, so proper regulation would definitely prevent excess urine loss, and also help to control toxin buildup in the kidneys. Now, the kidney use two mechanisms to achieve this renal autoregulation. Two mechanisms. The kidney uses two mechanisms to achieve this renal autoregulation. Two mechanisms. The two mechanisms, we have myogenic mechanism, and then we have tubuloglomerular feedback. Okay, So the kidney uses two mechanisms to achieve this renal autoregulation. We have the myogenic mechanism and then the tubulo-glomerular feedback. Now, with the myogenic mechanism, anytime your glomerular filtration rate is too high, is that okay? What will happen is that the afferent arterioles, okay, we have some cells within the afferent arterioles, the messenger cells. We have these cells would actually detect the high blood pressure. Is that okay? So anytime the glomerular filtration rate is very high, it will be detected in the afferent arterioles. So the afferent arterioles would now come in, okay, with constriction. They will constrict, okay, and when constriction occurs, it will minimize the flow of blood in the glomerulus. Okay, it will minimize the flow of blood in the glomerulus. Is that okay? And that would contribute to, reducing the glomerular filtration rate. It will slow down the amount of blood that moves into the glomerulus, or it will reduce the glomerular filtration rate. Is that okay? Good. So that is the afferent arteriole. Okay, and in a situation where your glomerular filtration rate is too low, it means that the renal blood flow towards the glomerulus, okay, is also very low. So that means that the glomerular filtration rate is very low. In that situation, the afferent arteriole will dilate, and the moment it dilates, it will allow a lot of blood to move towards, or to move into the, it will allow a lot of blood to move into the, it will facilitate the movement of a lot of blood into the afferent arteriole, and that would cause the glomerular filtration rate to increase. Okay, that process is what we call the myogenic mechanism. So anytime your glomerular filtration rate is very low, the afferent arteriole will constrict. And when the glomerular filtration rate, anytime your glomerular filtration rate is very high, the afferent arteriole will constrict. When it constricts, it will help to slow down the glomerular filtration rate. And anytime your glomerular filtration rate is very low, the afferent arteriole will dilate. When it dilates, it will facilitate the movement. A lot of blood will be able to, because we are talking about arterioles. Okay, so anytime your glomerular filtration rate is very low, then the afferent arteriole have to now dilate. But when your glomerular filtration rate is very high, then the afferent arteriole would have to constrict so that it will slow down the blood to the glomerular, the blood into the glomerulus, and that will help to reduce the glomerular filtration. That process is what we call the myogenic mechanism. The myogenic mechanism. Then we have the tubulo-glomerular feedback. The tubulo-glomerular feedback. Okay, the tubulo-glomerular feedback. Okay, now with the tubulo-glomerular feedback, what happens with the tubulo-glomerular feedback is that, okay, we have in the distal convoluted tubule, remember we mentioned that we have an area in the kidney we call the juxtal glomerular apparatus. Over here, I told you that the distal convoluted tubule has contact with the afferent arteriole. In this distal convoluted tubule here, in the distal convoluted tubule, we have some cells inside. We call it the macular denser cells. The macular denser cells. The macular denser cells. Okay, they are within this distal convoluted tubule. And we have some cells in the afferent arterioles. We call those cells the juxtal glomerular cells. The juxtal glomerular cells. Okay, take note of that. Now let's go back and continue. Now, anytime, okay, there's macular denser cells in the afferent arteriole. The macular denser cells, there's macular denser cells over here. They are within the afferent arteriole. Sorry, they are within the distal convoluted tubule. Okay, within the distal convoluted tubule. There's macular denser cells. What they do is that they are able to detect high sodium levels, or they are able to detect sodium levels. Let's keep it that way. That's the best. They are able to detect sodium levels. Okay, so listen, in a situation where your glomerular filtration rate is very high, it means that because there is fast movement of filtrate or fluid, a lot of sodium will reach the distal convoluted tubule. So the distal convoluted tubule, because the macular denser cells only detect sodium, it will detect the high sodium levels. So in effect, understand that when there is high sodium levels, Okay, it just can also be interpreted that there is high glomerular filtration rate. So the moment the macular denser cells detect the high sodium levels, is that okay? The high sodium levels is just the same as high glomerular filtration rate. Okay, so it will send a signal to the juxtaglomerular cells in the afferent arterioles. Then the juxtaglomerular cells, what they will do is that they are going to cause what we call constriction. Okay, they will cause what we call constriction. Is that okay? So when they cause constriction, because that is going to activate renin. Is that okay? It is going to activate renin. Okay, because when what happens is that when the macular denser cells actually detect the high sodium levels, they send a signal to the juxtaglomerular cells in the afferent arteriole. Then the afferent arteriole will now activate renin. Do you get it? Then you know that the end product of renin release would activate the renin-angiotensin-aldosterone cascade. Then that cascade would, in the end, lead to vasoconstriction. Okay, it will cause vasoconstriction in the afferent arterioles. Then the vasoconstriction would help slow down the glomerular filtration rate. Okay, in a situation where the glomerular filtration rate is high. I told you that when the glomerular filtration rate is very high, okay, that is when the macular denser cells is going to detect high sodium levels. Okay, when the glomerular filtration rate is very high, it means that more filtrate will reach the distal convoluted tubule, and the macular denser cells will interpret that as high sodium levels. Okay, and they will send a signal to the juxtaglomerular cells. Then the juxtaglomerular cells will activate renin release. So when renin is released, then renin release will also activate the renin-angiotensin-aldosterone cascade. And in the end, renin-angiotensin, when it is activated, in the end, it will cause vasoconstriction. Okay, then the vasoconstriction would help to slow down the blood movement in the arterioles. It will slow it down. Do you get it? It will slow it down so that will help the glomerular filtration rate to now reduce. Now, if the macular denser cells, on the other hand, detects what we call low sodium levels, low sodium levels can also be interpreted as low glomerular filtration rate. It means that the movement of the filtrate is too slow, so a little sodium is reaching the distal convoluted tubule. Okay, but the macular denser cells only detect the sodium, not the flow. Okay, but then if it detects low sodium level, it means that there is a low glomerular filtration rate. So it will send a signal. Once it detects low sodium level, it will send a signal to what? It will send a signal to the it will send a signal to what we call the it will send a signal to the juxtaglomerular cells. Okay, and this time the juxtaglomerular cells is not going to release renin. Is that okay? Good. So when it releases the renin, and then it activates the renin-angiotensin-aldosterone system, okay, at the end of the day, it is going to result in what? Afferent arterial vasodilatation. The afferent arterioles will rather what? Dilate. Is that okay? Good. Then again, another thing that will happen is that the afferent arterioles will dilate, okay, and then the efferent arterioles will rather constrict. Okay, so the afferent arterioles will rather dilate, then the efferent arterioles will rather what? Constrict. Is that okay? Because of the renin release from the juxtaglomerular cells. And in that situation, that would help to what? To increase the renal blood flow. Okay, so the renal autoregulation is like a mechanism that the kidney uses to what? To regulate your blood flow, or the renal blood flow in the kidney, so that at the end of the day there wouldn't be a high glomerular filtration rate or a low glomerular filtration rate. Okay, so in summary, anytime with a tubuloglomerular feedback, okay, let me talk about the myogenic mechanism. So for the myogenic mechanism, that is pretty simple. So if your glomerular filtration rate is very high, the afferent arterioles will constrict. If the glomerular filtration rate is very low, the afferent arterioles will dilate. Okay, now with the tubulo-glomerular feedback, we have structures like the macular denser cells in the distal convoluted tubule and the juxtaglomerular cells in the afferent arterioles. So when your body detects when your macular denser cells in the distal convoluted tubule detects high sodium levels, you should interpret that as high glomerular filtration rates. Okay, and then once there is high glomerular filtration rate, what is going to happen is that it is going to send a signal to the juxtaglomerular cells, and the juxtaglomerular cells, what it will do is that the juxtaglomerular cells will now reduce it will release what we call vasoconstrictive signals. And these vasoconstrictive signals would cause the afferent arterioles to constrict. Okay, and that would help to reduce the flow, okay, of blood through the afferent arterioles into the glomerulus, which will help to reduce the glomerular filtration rate. On the other hand, if the macular denser cells rather detect low sodium levels, okay, the low sodium levels as low glomerular filtration rate. So if there is low glomerular filtration rate, it's like you are having a low blood pressure. So the renin-angiotensin-aldosterone system will be activated, because you remember that renin-angiotensin-aldosterone system is activated when your blood pressure is low. Okay, so once the renin-angiotensin-aldosterone system is activated, renin will first be released. Then it will convert angiotensinogen into angiotensin 1. Then in the end of the cascade, okay, vasoconstriction would occur. Okay, so there is going to be specifically there is going to be vasoconstriction in there is going to be vasoconstriction in the efferent arterioles, and there is going to be vasodilation in the afferent arterioles, and that would help to increase the glomerular filtration rate. So basically, this is what we call the renal autoregulation. Renal autoregulation is very simple. Renal autoregulation. So this diagram is showing the renal autoregulation. Okay, so we have the myogenic mechanism, and then we have the tubulo-glomerular feedback mechanism. The myogenic mechanism, and then the tubulo-glomerular feedback mechanism. Okay, good. And this relies on inherent properties of the arterioles. Is that okay? Good. So basically, this is it. So if you look over here with the myogenic, if the glomerular filtration rate increases, that means the renal blood flow has increased. Okay, the hydrostatic pressure against the walls of the afferent arterioles will increase. So the smooth muscle walls, we have some stretch receptors in the smooth muscle walls of the afferent arterioles. They will detect the increase. Okay, and then they begin to release vasoconstrictive signals, and then at the end, the afferent arterioles will now constrict. So you can see the constriction over here, that the afferent arteriole compared to this has now constricted, and that will help to decrease the renal blood flow and decrease the glomerular filtration rate. So you can see that in the end, because of the mechanism, the myogenic mechanism, the renal blood flow has reduced, and then the glomerular filtration rates have also reduced. Do you get it? And then the pressure within the glomerular capillaries have also reduced. Is that okay? Good. Now, if you come over here, okay, the tubulo-glomerular feedback. Okay, that one has got to do with the macular denser cells, which detect the sodium levels. So if the sodium levels are high, the macular denser cells will detect the high sodium levels. Okay, and they will send a signal to the juxtaglomerular cells. Is that okay? And that will cause the release of constrictive substances. Okay, constrictive signals. Then in the end, the afferent arterioles will constrict. Okay, but then if it detects low sodium levels, it's just the same as it's detecting, or it's just the same as there is low glomerular filtration rate, which can also be interpreted as low blood pressure. And you know, low blood pressure is what activates renin-angiotensin-aldosterone system. So when renin-angiotensin-aldosterone system is activated, in the end, it causes vasoconstriction, but that vasoconstriction would have its impact on the efferent arteriole. Okay, but then there would be vasodilation in the afferent arteriole, and that will cause the renal blood flow to increase. You need to understand it. It's very simple. That will cause the renal blood flow to increase, and in the end, okay, the glomerular filtration rate would also increase in a situation where there was an increase in sodium levels, okay, was detected by the macular denser cells. So basically that is renal autoregulation. Okay, now this slide is showing substances and the glomerular filtration rate. So on our right, we have substances that can cross the glomerular filtration membrane. And on our left, we have substances that cannot cross the glomerular filtration membrane. So we have substances that can cross the glomerular filtration membrane. And we have substances that cannot cross the glomerular filtration membrane. With the substances that can cross the glomerular filtration membrane, we have water, we have ions, we have glucose, we have small molecules, and then we have small proteins and peptides. These are all substances that can cross the glomerular filtration membrane. We have water, we have ions, we have glucose, we have small molecules, and we have small proteins and peptides. These substances can all cross the glomerular filtration membrane. Then on our left, these are substances that cannot cross, mostly because some of these substances are large, like large proteins like albumin and other large proteins. Okay, we have blood cells like red blood cells, white blood cells, and platelets because of their size. Then we have large molecules like lipoproteins and other complex molecules. Then we have negatively charged molecules. Okay, the reason is because the glomerular basement membrane of the glomerular filtration membrane is negatively charged. Is that okay? Good. So negatively charged molecules also cannot cross the glomerular filtration. So basically, this is it for substances in the glomerular filtration membrane. So these substances on our left cannot cross the glomerular filtration membrane, but these substances can cross the glomerular filtration membrane. All right, so I've explained the juxtaglomerular apparatus. So the juxtaglomerular apparatus is the point where the distal convoluted tubule actually comes in contact with the afferent arteriole. Okay, so what components form the juxta? So you can be asked this in exams, in anatomy exams. So the components that forms the juxtaglomerular apparatus, we have the distal convoluted tubule and then the afferent arteriole. So within the distal convoluted tubule, we have the macular denser cells. Okay, and within the afferent arteriole, we have the juxtaglomerular cells. They are within the walls of the afferent arterioles. Okay, and they basically act as baroreceptors and secrete renin in response to low blood pressure and low sodium levels, and also activates sympathetic stimulation. Is that okay? So in a situation where there is low sodium levels, the signal of low sodium levels are sent, renin will be activated, or low blood pressure, renin will be activated, and that will increase blood pressure and also sodium reabsorption. And at the end of the day, glomerular filtration rate will increase. Is that okay? So basically that is it. So in high sodium levels, afferent arterioles will constrict. In low sodium levels, signals the juxtaglomerular cells to release renin. Then in the end, glomerular filtration rate would increase. Now together, the juxtaglomerular cells and the macular denser cells regulate glomerular filtration rates and maintain blood pressure via the tubuloglomerular feedback and hormonal control. The relevance is that this is very crucial in blood pressure regulation because dysfunction may contribute to hypertension and renal pathologies. Okay, so basically that is it. Okay, basically that is it. Now, we mentioned of the fact that the kidney plays a role in the production of red blood cells. On this slide, we are going to learn about how the kidney is able to perform that role. Within the renal cortex of the kidney and the renal medulla, we have some cells over there we call the peritubular fibroblast-like cells. Peritubular fibroblast-like cells. In fact, these cells are within the renal cortex and then the outer medulla of the kidney. And these cells produces erythropoietin. So now, in a situation where the oxygen levels in your blood are very low, okay, these cells would be stimulated to produce what? Erythropoietin, their hormone. We call it EPO, renal EPO-producing, or renal EPO hormone. So they produce this erythropoietin. Then this erythropoietin actually acts on the bone marrow. Is that okay? For the bone marrow, through a process we call erythropoiesis to produce a lot of red blood cells, and that will increase your blood oxygen carrying capacity. Okay, good. So we call those cells peritubular capillaries. For anatomy, it's very essential that you know where these cells are located. Peritubular like cells, they are located within the renal cortex and then the outer medulla of the kidneys. Okay, and they produce the erythropoietin. Okay, so it's essential that you know where exactly those cells are within the renal cortex and then the outer medulla of the kidney. They produce the erythropoietin or EPO. Okay, EPO. Okay, so if your blood oxygen levels are very low, okay, if your blood oxygen levels are very low, is that okay? Good. When your blood oxygen levels are very low, these cells are actually activated, then they produce erythropoietin. Then the erythropoietin will also act on the bone marrow, okay, to produce more red blood cells via a process called erythropoiesis. Then your oxygen levels or oxygen carrying capacity of the blood will now increase. Okay, so basically that is it for. But then it uses this kind of mechanism, okay, a negative feedback mechanism. So when it increases, then at the end of the day, the release of this erythropoietin will now decrease. Is that okay? Because if your oxygen carrying capacity increases, there will not be any need for an increase in the production of the erythropoietin. So it uses a negative feedback mechanism to regulate this release. Okay, now let's do the urine formation. The urine formation or formation of urine. Now, urine formation is essential for removing waste and maintaining fluid and electrolyte balance. It occurs in 4 stages. The 4 stages, we have filtration, we have reabsorption, we have secretion, and then we have excretion. Now, let's look at the structures involved from the diagram. Let's go through the process of urine formation from the diagram. The first process of urine formation is filtration. And filtration occurs over here in the glomerulus. It occurs in the glomerulus of the nephrons. Okay, so with the filtration, the blood is filtered through the glomerular membrane. This is the glomerular membrane, the glomerulus. The blood is filtered through the glomerular membrane into the Bowman's capsule. That process is what we call filtration. That is the first process of urine formation, whereby blood is filtered through the glomerular membrane into the Bowman's capsule. So once the blood gets here, we don't call it blood, we call it filtrate. Okay, that is what we call filtration. Okay, so filtered substances like water, salt, glucose, amino acids, and waste like urea, all will be found over here within the filtrates. Okay, then because you see, for example, imagine substance like water. Water can freely pass through the membrane. So imagine all your water is here and they go out to form part of your urine. You dehydrate and can even go into shock and die. That is why we have the second process we call reabsorption, so that there will be some checks and balance, so that all the essential substances that were freely filtered, they will be reabsorbed back into the blood with the help of the peritubular capillaries, and that is what we call reabsorption. Reabsorption mostly occur in the proximal convoluted tubules, in the loop of Henle, in the distal convoluted tubules. Okay, so reabsorption mostly occur in the proximal convoluted tubule, but it can also occur in the loop of Henle and then the distal convoluted tubule. In fact, some substances may even find their way out and they may not be filtered. They can be secreted back. Secretion also have got to do with waste that, for some reasons they were not able to be filtered, or toxins that are within the blood, they were not able to be filtered, where they are secreted back into the tubule. That is what we call the secretion. So the thing is about checks and balance. So the blood will check, is a certain electrolyte or substance balance, or is it in excess in the blood? If it's in excess, then they will be released into the tubules. In the tubules too, there are some checks. If a certain electrolyte is in excess, then they will be released into the peritubular capillaries or the blood. Is that okay? So it's about checks. So the secretion mostly occurs in the distal convoluted tubule and collecting duct. Okay, whereby additional waste like hydrogen ions, potassium ions, bicarbonate, okay, are usually added from the blood depending on your body's electrolyte and acid base balance or levels. Okay, that's the secretion. Then the last process is excretion. Okay, so from the collecting that, it's now a urine, so it moves to the ureters, into the urinary bladder, in through the urethra. Then finally the urine is expelled out of the body. Okay, this process is what we call urine formation. Okay, so it involves 4 processes: filtration. And if you are describing formation of urine, you are not able to mention these 4 processes, your description is not complete. So this urine formation has 4 processes: the filtration, reabsorption, secretion, and then excretion. Now remember that the kidney receives approximately 20 to 25% of your cardiac output. The kidney receives approximately 20 to 25% of your cardiac output. So in effect, we do this calculation over here. Excretion is usually equal to filtration minus reabsorption plus secretion. Okay, because you see, we have an outward, process. The filtration is an outward process. Okay, and then the reabsorption is also what? An inward process. The reabsorption is an inward process, it's negative. Okay, and secretion is also more like an outward process, which is also positive. Okay, so if you want to calculate the excretion, the final amount that will be excreted, or the urine, okay, that will be excreted, is the filtration, the amount of the filtrate minus the amount of the filtrate that is reabsorbed plus the amount that is secreted back from the peritubular capillaries. So the filtrate minus the reabsorption plus the secretion, then that will give you the excretion. Okay, good. So this diagram is also explaining the urine formation. So the first process begins over here in the glomerulus. Okay, so this is the afferent artery which brings the blood inside. Then it is filtered into the proximal convoluted tubule. Then over here we have the filtrate. Then you see we have the peritubular capillaries going around the nephron. So within the proximal convoluted tubule, reabsorption takes place. So you see the arrow is pointing from the proximal convoluted tubule into the peritubular capillaries. So there is reabsorption over here. Then reabsorption can also occur in the descending limb and then the ascending limb of the loop of Henle. So you see the arrow is moving from here to the blood vessel. So it means that reabsorption. Okay, and then if you come over here, you can see now the arrow is now moving from the peritubular capillaries into the tubule, and that is what we call the secretion, whereby essential substances are secreted from the blood into the tubule. Okay, then finally they end up in there. But even within the collecting duct, okay, there can be some reabsorption of water, especially under the influence of antidiuretic hormone from the brain. There can be some reabsorption of water. In fact, there is always what we call reabsorption of sodium. Okay, reabsorption of sodium, especially under the influence of aldosterone. At the levels of the distal convoluted tubule, there can be reabsorption of sodium. Okay, if renin-angiotensin-aldosterone system is activated, there can be some level of reabsorption of sodium, especially when the distal convoluted tubule detects low levels of sodium. Okay, aldosterone will be activated via the renin-angiotensin-aldosterone system, and then aldosterone release will cause reabsorption of sodium into the peritubular capillaries. Okay, so basically this is what we call the urine formation. So we have 4 processes: filtration, reabsorption, secretion, and then excretion. Okay, so once it hits over here, then we have excretion. So in summary, the kidney plays a role in excretion of waste. It removes nitrogenous waste like urea, creatinine, and uric acid from the body. It regulates electrolyte levels. It regulates blood pressure. It helps to maintain acid-base balance. It plays a role in erythropoiesis regulation. It also helps to regulate fluid volume. It plays a role in detoxification, and then it also plays a major role in vitamin D synthesis, whereby it helps to convert inactive vitamin D to calcitrol, which is the 125 dihydroxycholecalciferol. Is that okay? Great. So basically, these are the functions of the kidney. Now, Let's look at the renin-angiotensin-aldosterone system. The renin-angiotensin-aldosterone system. Kindly pay attention. This system sometimes can confuse students, even though it's very simple mechanism. The renin-angiotensin-aldosterone system. Now, the renin-angiotensin-aldosterone system, which we sometimes refer to as the RAS, okay, is a hormonal system that plays a critical role in regulating your blood pressure, fluid and electrolyte balance, as well as systemic vascular resistance. So it's a hormonal system, meaning the process or the mechanism involves the activity of some hormones in your body. Okay, now it is activated when your body detects a drop in blood pressure or a decrease in sodium levels, triggering a cascade of events that helps restore normal conditions. Now, this system involves the release of hormones like renin, angiotensin 2, and aldosterone that work together to increase blood pressure and regulate kidney function. Okay, good. Now, what are the importance of the renin-angiotensin-aldosterone system? We would get to the mechanism itself. We are looking at some preliminaries before we start with the mechanism. Now, what is the relevance of the renin-angiotensin-aldosterone system? The relevance is that it regulates blood pressure. Okay, so the renin-angiotensin-aldosterone system is very essential for maintaining long-term blood pressure control by preventing hypotension, low blood pressure, and maintaining adequate blood flow to organs. Okay, it also plays a role in fluid and electrolyte balance. It helps you to respond to stressful situations like dehydration, hemorrhage, shock, which helps to restore blood volume and pressure. Now, the clinical significance is that if there is a malfunction of this renin-angiotensin-aldosterone system, it can lead to diseases like hypertension, heart failure, and chronic kidney disease, and that makes it a key target for a lot of therapies in the field of pharmacology. Is that okay? We have a medicine we call the angiotensin converting enzyme inhibitors and angiotensin 2 receptor blockers, which you learn all these in pharmacology. Okay, so quickly, let's go to the renin-angiotensin-aldosterone system. Now, these 5 organs, okay, or structures are all involved. These are the organs that are involved in the renin-angiotensin-aldosterone system. First of all, you need to know the organs or the structures that are involved in this renin-angiotensin-aldosterone system before you be able to understand the mechanism or the system. You should know all the organs that are involved. So first of all, the kidney is involved in this process. The liver is involved in this process. The lungs are involved in this process. The adrenal glands are involved in this process. And blood vessels are involved in this process. So if you are explaining the mechanism or the system and your explanation does not address the functions or the role played by all these 5 organs, it means your explanation is not complete. Okay, so the kidney, let's look at the role of the kidney. Now, the kidney is the first organ that plays a key role in this renin-angiotensin-aldosterone system. Okay, so the renin-angiotensin-aldosterone system is actually activated when there is a low blood pressure or low blood sodium levels. Okay, good. This is the triggering factor for renin-angiotensin-aldosterone system. So if you are asked in exams, what actually triggers the renin-angiotensin-aldosterone system? When there is a low blood pressure or low blood sodium levels. Okay, so anytime there is a low blood pressure or low blood sodium levels, your kidney will release renin. This is the first enzyme in the RAS cascade. If you are explaining renin-angiotensin-aldosterone system, you are not able to talk about the renin release first. You've deviated. Okay, so anytime your blood pressure is very low or blood sodium levels are very low, the kidney will release renin, which is the first enzyme in the process. Now, after renin has been released, already your liver produces angiotensinogen, which is a protein. Okay, and this renin, which has been released by the kidney because of low blood pressure, it will convert this angiotensinogen into angiotensin 1. Okay, good. Now, this angiotensin 1 is a less potent or an inactive vasoconstrictor. It's a less potent vasoconstrictor, even though it's a vasoconstrictor anyway, but it is less potent. Okay, now this angiotensin 1 would keep on circulating in the blood until it gets to the lungs. In the lungs produces an enzyme called the angiotensin converting enzyme, and this ACE, angiotensin converting enzyme, will convert angiotensin 1 to angiotensin 2, and this angiotensin 2 is a potent vasoconstrictor. Is that okay? Now, this angiotensin 2 actually stimulates the adrenal gland for the adrenal gland to release aldosterone, and the aldosterone can act on the distal convoluted tubule to facilitate reabsorption of what? Sodium. And as more sodium is being reabsorbed, you know, anywhere sodium goes, water follows. Water follows, so that will increase the blood volume. As your blood volume increases, at the end of the day, what is going to happen is that your blood pressure would also what? Eventually increase. Is that okay? Good. And another thing is that this angiotensin 2, okay, can also stimulate sympathetic stimulation. It will activate sympathetic stimulation, and sympathetic stimulation will cause vasoconstriction. Okay, it will narrow blood vessels, and that would also what? Increase systemic vascular resistance, which would help to increase your blood pressure. Okay, this is what we call the renin-angiotensin-aldosterone system. So the organs involved, we have the kidneys, we have the liver, we have the lungs, we have the adrenal glands, and the blood vessels. Now let's look at this same renin-angiotensin-aldosterone system in a diagram. Good, let's look at this diagram. So like I said, the renin-angiotensin-aldosterone system is a system that is activated anytime there is a decrease in your blood pressure, or when the blood flow to your kidneys actually decrease. So anytime the blood flow to your kidneys decreases, the kidney first of all releases renin, which is an enzyme. It releases this renin. Okay, now already in your blood, the liver produces and releases angiotensinogen into the blood. This is a protein. It releases this protein into the blood, angiotensinogen. Okay, now because renin has been produced, renin will come and meet this angiotensinogen and convert angiotensinogen to angiotensin 1. Now, this angiotensin 1 is not a potent vasoconstrictor, even though it is a vasoconstrictor, but not potent. And so it needs to be activated to become potent. So the angiotensin 1 is also in the blood now. It keeps on circulating until it gets to the lungs. Then the lungs, which also produces angiotensin converting enzyme, will produce its enzyme, angiotensin converting enzyme, to convert the angiotensin 1 to angiotensin 2. And this angiotensin 2 is a potent vasoconstrictor. And this same angiotensin 2 can also act on the kidney for the kidney to reabsorb a lot of sodium. Okay, this angiotensin 1 can also act on the brain, send a signal to the brain for the brain to release more antidiuretic hormone. And antidiuretic hormone would automatically prevent loss of water for you to retain a lot of water. So in effect, the aldosterone release and then the antidiuretic hormone release would all cause fluid retention and sodium retention. So sodium levels will increase, fluid levels would also increase. Then that would help to increase your blood pressure. Apart from that, this angiotensin 2 also stimulates sympathetic release or sympathetic activation. And once sympathetic system is activated, vasoconstriction will occur in the peripheral blood vessels. So the peripheral blood vessels will constrict, so vasoconstriction will occur. So the increase in blood sodium levels coupled with the retention of fluid or fluid retention, okay, plus the vasoconstriction, all would lead to increase in blood pressure. Okay, this mechanism is what we call the renin-angiotensin-aldosterone system. The renin-angiotensin-aldosterone system. So in summary, anytime there is a decrease in your blood pressure, the blood flow to your kidneys decreases, or glomerular blood flow decreases, the kidney will release this protein, this enzyme we call renin. Already in the blood, the liver produces angiotensinogen. So angiotensinogen will also meet up with renin. Renin will convert angiotensinogen into angiotensin 1. Then angiotensin 1, okay, keep circulating in the blood. It's a vasoconstrictor, but it is not potent. Okay, it keeps circulating until it reaches the lungs, and the lungs releases an enzyme called angiotensin converting enzyme, which converts the angiotensin 1 to angiotensin 2. Then the angiotensin 2 1, it activates sympathetic stimulation. And once sympathetic system is stimulated, vasoconstriction will occur in the peripheral vessels. It also acts on the adrenal gland to release aldosterone, and aldosterone acts on the kidney to reabsorb more sodium and water. It also sends a signal to the brain to release more antidiuretic hormone, and antidiuretic hormone will stop the formation of urine. It's an antidiuretic, so it will pause the formation of urine, so more fluid will be retained. So in effect, the sodium levels will increase, a lot of fluid will be retained, and then the sympathetic stimulation will also cause vasoconstriction. Then all these will lead to increase in blood pressure. This is a summary of the renin-angiotensin-aldosterone system. Okay, good. This is a summary of the renin-angiotensin-aldosterone system. So this is a summary. So renin release in the kidney. So when blood pressure drops or sodium levels are low, the kidney secretes renin into the bloodstream. So angiotensinogen activation in the liver. So the renin converts angiotensinogen from the liver into angiotensin 1. Then angiotensin converting enzymes in the lungs now converts the angiotensin 1 into angiotensin 2. Then the effect is that there will be vasoconstriction because of the sympathetic stimulation, whereby there is going to be narrowing of the blood vessels, which will raise the blood pressure. Now, it will also act on the adrenal glands, stimulate the adrenal glands to secrete aldosterone, which increase sodium and water reabsorption by the kidneys, further raising the blood pressure. Now, then again, angiotensin 2 also triggers the release of antidiuretic hormone, okay, enhancing water retention. So basically, that is it. In fact, if your renin-angiotensin-aldosterone system is not regulated properly, you can end up with hypertension, you can end up with heart failure, you can end up with a kidney damage, edema or fluid retention, electrolyte imbalance. It can increase your risk of stroke and heart attack. It can lead to atherosclerosis, and then the last is it can lead to metabolic disorders. Okay, and this is why the renin-angiotensin-aldosterone system is very essential. Now, let's look at the other organs of the urinary system. So all the things that we've been doing for the past 1 hour, we've been looking at the kidneys. Now, with the ureters, the next organ of the urinary system is the ureters. In fact, the ureter is a muscular organ, and it's also retroperitoneal. Okay, it's a retroperitoneal tube which is approximately 25 centimeters long and 1.7 centimeters wide near the bladder. Okay, so the ureter is a muscular organ. It's also retroperitoneal tube. It's a tube behind the peritoneum, approximately 25 centimeters long and 1.7 centimeters wide near the bladder. So this is what we are looking at, the ureter. This one, the ureter. Remember the anatomical description. It is approximately 25 centimeters long, 1.7 centimeters wide, and it is near the bladder. Now, this ureter actually transports urine from the renal pelvis into the urinary bladder. It transports urine from the renal pelvis over here into the urinary bladder. Okay, good. Now, in terms of the pathway, it descends the peritoneum, cross the pelvic brim. This is the pelvic brim. It crosses the pelvic brim and then enters the bladder posteriorly. Okay, posteriorly and obliquely at the trigon area. So we have the uteric orifice over here. We have another uteric orifice over here. Then the urethral orifice is also over here. So this orifice 1, 2, 3 forms a triangular, demarcation over here that we call the trigon of the bladder. We will look at that later on. Okay, in terms of backflow, there is a mechanism to prevent backflow in the ureters. Okay, now this mechanism is that as the bladder pressure increases, the urethral openings are compressed. Anytime the bladder pressure in the bladder increases, okay, as it keeps filling, the urethral openings are compressed, and that prevents reflux of urine back into the kidneys. Now, in terms of its wall structure or structure of this ureter, okay, it has 3 layers. The 3 layers, we have the outermost layer, which is a fibrous coat, which we also call the adventitia. We have the innermost layer, which is a muscle coat. okay, which we also call the muscularis, and then we have the innermost layer, which is called the mucosa. Okay, so these are the layers of the ureters: the adventitia, the muscularis, and the mucosa. The adventitia is the fibrous connective tissue anchoring the ureters to surrounding structures. So we also call it the fibrous coat, which is the outermost. We have the middle layer, which is made up of two layers of longitudinal and outer circular smooth muscles. Okay, and this layer, that is the layer that initiates peristaltic contraction every few seconds to minutes, depending on urine flow. And then we have the innermost layer, which is the mucosa, made up of transitional epithelium with a narrow lumen, continuous with the renal pelvis above, and then the bladder below, susceptible to obstructions. Okay, now let's do the urinary bladder. The last but one, the urinary bladder. Okay, the urinary bladder. The urinary bladder is actually a hollow, distensible muscular sac located in the pelvic cavity, posterior to the pubic symphysis and inferior to the peritoneum. It is just behind the pubic bone in front of the rectum in males and the uterus in females. It stores urine until it is expelled from the body. The capacity is that it typically holds about 500 ml of urine and can expand to about 700 to 800 ml. Okay, so this is what we call the urinary bladder. In fact, it is made up of muscles. Okay, and you know, once it is made up of muscles, it tells you that it can contract and it can relax. You know, muscles have the ability to contract and they also have the ability to relax. In fact, the muscles that we have in the urinary bladder, we call it the detrusor muscles. And it is very important that you understand because it has some physiologic relevance when it comes to micturition. Okay, detrusor muscles. Okay, so that's the muscles that forms the urinary bladder, the detrusor muscles. Okay, so we said that it's located in the pelvic cavity. Okay, posterior to the symphysis, pubic symphysis, and inferior to the peritoneum. And it is just behind the pubic bone and in front of the rectum in males. Okay, and the uterus in front of the rectum in males and then the uterus in females. And we said that it stores urine until it is expelled from the body. The capacity is that it can typically hold about 500 ml, but then it can expand to hold 700 to 800 ml of urine, or it can expand to a volume or a capacity of about 700 to 800 ml. Okay, now the outermost covering of the urinary bladder is the parietal peritoneum. Okay, just that the layer of this parietal peritoneum, the urinary bladder is just the same as what the layers of the urinary bladder. Okay, so we have the adventitia, which is the fibrous coat. We have the middle layer, which is the muscularis, and then the mucosa. Okay, the layers are just the same. Okay, so we have the walls or layers. The bladder consists of the detrusor muscles, which are 3 layers of smooth muscles, and then the mucosal lining of transitional epithelium with rugae for expansion. Okay, because anywhere we have rugae in the mucosa, rugae will allow for expansion of that very tissue or cavity or layer. Okay, so we have the fibrous adventitia, just the same as we have the fibrous adventitia covering the ureters. We have the same thing covering it. Okay, good. Just that the urinary bladder is first of all, the outermost is the parietal peritoneum. Is that okay? Good. Then we have the uteric orifice. These are the uteric orifice. So you see the ureters are here. So the ureter enters the bladder posteriorly through the uteric orifice. So we have the uteric orifice over here, and we have uteric orifice over here. The uteric orifice, and then we have another uteric orifice over here. Okay, the uteric orifice. So the uteric orifice are 2 because we have 2 ureters. The uteric orifice are also 2. Okay, they are the 2 small openings located at the posterolateral corners of the bladder triangle. Okay, so this uteric orifice and this uteric orifice, and then we have the urethral opening. Okay, these three things, you see, it forms a triangle. So from here to here, from here to here, okay, it forms a triangle like this. And that is what we call trigon of the urinary bladder, or trigon of the bladder. Okay, trigon of the bladder. Okay, more like a smooth triangular region defined by the openings of the two ureters and then the urethra. Okay, and it's a common site for infection. It's a very common site for infection in males and females, or in the urinary system. Okay, now, okay, over here, so for males, okay, over here, just below the urinary bladder, we have a sphincter over here. We call it the internal urethral sphincter. And then from the internal urethral sphincter, from here to this side, we have the prostate gland. And then just under the prostate gland, we have the external urethral sphincter also over here. But then just under the prostate gland, we have the urogenital diaphragm. It's not here, it's not shown here in this diagram, but I will show you another diagram which have those features. Is that okay? Good. So we have the internal urethral sphincter, which is an involuntary muscle that automatically keeps the urine in the bladder. It works automatically. Okay, then we have the external urethral sphincter, which work voluntarily. This is what helps you to decide that you are going to urinate. Is that okay? Good. Now, the final thing we are going to look at is the urethra. The urethra. That is the final thing that we are going to look at. Okay, the urethra. Now, the function of the urethra is that it conveys urine from the bladder to the outside of the body. That's the function of the urethra. This is what we call the urethra. The urethra. It conveys urine. You see, it has been labeled over here, the urethra. The urethra. It conveys urine from the bladder to the outside of the body. In females, it is very short. It is about 3 to 4 centimeters long. Okay, and it is bound to the anterior vaginal wall. Now, the external orifice of the urethra is, in females, is located between the vaginal opening and then the clitoris. We will look at that when we are doing the reproductive system. Now, in males, it is very long, and it is divided into 3 regions. We have the prostatic urethra, which is about 2.5 centimeters. We have the membranous urethra, which is about 0.5 centimeters. And then we have the spongy or penile urethra, which is about 15 centimeters. Okay, now this is the prostatic urethra. So you see the urethra starts from here to this point. The area that spans within the prostate gland, that is what we call the prostatic urethra. So from this side to this side is the prostatic urethra. Then we have the membranous urethra. The membranous urethra is the part of the urethra that passes through the urogenital diaphragm. This is what we call the urogenital diaphragm. The urogenital diaphragm is over here. This area is what we call the urogenital diaphragm. Okay, that part of the urethra that passes through the urogenital diaphragm is what we call the membranous urethra. Okay, good. And then we have the spongy urethra, or the penile urethra. That is the part that passes through the corpus spongiosum of the penis. This is what we call the corpus spongiosum. We have the corpus spongiosum. From here up to here is the corpus spongiosum. We have some also here. From here up to here, that is the corpus spongiosum. So from this side to this side, okay, the urethra that's come from here to here is what we call the penile or spongy urethra. In fact, the longest part of the male urethra is the spongy or penile urethra. It's about 15 centimeters. Is that okay? Then from the spongy or penile urethra, we have the next longest part of the urethra is the prostatic urethra, which is about 2.5 centimeters. Then the membranous urethra is about what? 0.5 centimeters. That is the shortest of all the segments of the male urethra. Okay. Now, transitions from the transitional near bladder to pseudo stratified columnar and then stratified squamous near the orifice. Okay, if you are looking at the inner lining of the urethra, there are changes depending on the layer where they are. Okay, so we have transitions from transitional epithelium, the transitional epithelium near the bladder, to pseudo stratified columnar and then stratified squamous near the orifice. Is that okay? Good. So that is just about the nature of the epithelium of the urethra, because in the bladder we said we have transitional epithelium, but it changes from the transitional epithelium near the bladder, and then as it comes inside, the inner lining becomes pseudo-stratified columnar. Then as it comes more deeper, it's stratified squamous near getting to the orifice, stratified squamous epithelium. Now with the sphincters within the urethra, okay, we have the internal urethral sphincter, and then we have the external urethral sphincter. The internal ones is not under your control, it's under involuntary control, but then the external one is a skeletal muscle and it's under voluntary control, and that is what you can control to decide that you are going to urinate. Is that okay? Good. Now let's look at one other very important thing in physiology. Okay, but before that, let's look at the blood supply, the lymphatic drainage, the nerve supply of the urinary system. Now for blood supply, for the kidney is supplied by the renal arteries, which are branches of the abdominal aorta and drained by the renal veins into the inferior vena cava. Now the ureters receive blood from the renal, the gonadal, common iliac, and then the vesical arteries. For the urinary bladder, it is supplied mainly by the superior and inferior vesical arteries, which are branches of the internal iliac arteries. For lymphatic drainage, for kidneys and ureters, they drain into the lumbar, which is the para-aortic lymph nodes. For the urinary bladder, they drain into the external and internal sacral iliac lymph nodes. For the nerve supply, okay, the kidneys are supplied by plexuses of sympathetic and parasympathetic fibers from the thoracic 10 and then the lumbar 1 vertebra. Okay, then for the ureters, they are innervated by the renal, aortic, hypogastric, and pelvic plexus. Then for the bladder, they are innervated by parasympathetic and sympathetic and somatic. So parasympathetic S2 and S4, they stimulate bladder contraction. Then the sympathetic T11 and L2, they promote bladder relaxation and internal sphincter contraction. Then for the somatic, the pudendal nerves, they play a role in control of external urethral sphincter, which is your voluntary control. That is why it's somatic or pudendal nerve. All right, so this is what we are talking about, the blood supply. So you can see the main aorta, it branches. So the renal artery, that is what supplies there. So a branch from the abdominal aorta, which goes to supply the renal artery, it branches into the renal artery. Okay, good. And then to the ureters, you see we have this one over here. So we have the common iliac vein, which also helps in the draining. We have the common iliac artery, which goes to supply the bladder. Okay, and then portions of the urinary bladder, and then portions of the urethra. Is that okay? Good. In terms of the nerve supply, if you have the pelvic nerve, which is sympathetic, we have the hypogastric nerve, which is parasympathetic, we have the pudendal nerve, which is somatic, and that is what innervates the external sphincter, like we said, the external sphincter for voluntary control, like we said earlier. All right, now let's look at the differences between the male and then the female urethra. The differences between the male and then the female urethra. Now, like we said earlier, the male urethra is relatively longer than the female urethra. The male urethra is between 18 to 20 centimeters, whilst the female urethra is between 3 to 4 centimeters. The other thing is that the male urethra performs dual function. It's a passageway for urine excretion, and it's also a passageway for semen. But the female urethra only have one role. It's only for urine excretion. Okay. Now, in terms of the location, the male urethra runs or spans through the prostate, the urogenital diaphragm, and the penis. But the female urethra, it doesn't pass through any prostate. It is just a straight path above the vagina. That is the reason why it is very short. Okay. Now, you remember that the male urethra has a lot of segments. It has about 3 segments: the prostatic urethra, the membranous urethra, and the spongy or penile urethra. But then the female urethra do not have any distinct segments. They are not into any divisions. Is that okay? Now, the tip of the male urethra is at the glans penis, whereas the tip of the female urethra is in the vulva between the clitoris and the vaginal opening. Okay, the male urethra is also associated with some structures like the prostate gland and the erectile tissues. That's the corpus spongiosum and all that. But then the female urethra is just associated with the urogenital diaphragm and then the sternal genitalia. Now, because of how long the length of the male urethra, okay, they are at low risks of developing urinary tract infections as compared to females. So in effect, females are at higher risks of developing UTIs as compared to males because of their short length and the proximity of, their perineal area to their anus. Is that okay? So because of they are at high risk of developing urinary tract infections. Now, with the sphincters, for the male urethra, they have both the internal and the external sphincter. So they have the internal sphincter, which is involuntary, and the external sphincter, which is voluntary sphincters. But then these sphincters are within, this one is just below the bladder. The internal one is below the bladder. Okay. That is before the prostate gland, and the external one is within the urogenital diaphragm. But for the female urethra, both the internal sphincter and the external sphincter are within the urogenital diaphragm. Is that okay? The significance is that the male ones are less prone to infection, but then because of the length of the male ones too, their urethra is prone to urethral strictures as compared. These ones are short, so they are rather prone to infections than urethral strictures. So female urethra is prone to urinary tract infections, whilst male ones are prone to urethral strictures because of the length. Okay, good. So these are the differences. So you see that for the males, okay, this is the male. You see we have the prostate gland between. This is the urethra. So from here to here is what we call the prostatic urethra. And over here is the membranous urethra. Because you see, this is the urogenital diaphragm. Okay, so this is the membranous urethra. Then from here to here is the spongy urethra or penile urethra. Is that okay? Then we have the external urethra orifice. This is the internal urethra orifice. Okay, then the sphincter is over here. The internal urethral sphincter is over here. The internal urethral sphincter is over here. This is the prostate gland. Then external urethral sphincter is also over here within the urogenital diaphragm. But for the females, both the external urethral sphincter and then the internal urethral sphincter, they are all within the urogenital diaphragm at this region. They are all within this one. That is what we were talking about. Is that okay? So relatively, you can see that the male urethra is very long. Very, very long. So here is 0.5. From here to here is 2.5. And then from here to here is 15. All would add up to 18 centimeters. And that's the reason why the male urethra is very long. And that's the reason why it's very prone to hot strictures. As compared, this is very short. So it's not prone to strictures. But because it's very short, okay, and because of the location of the urethral opening or orifice, they are prone to infections. Is that okay? They are high risk of getting urinary tract infections as compared to their male counterparts. Is that okay? Good. Now, the last thing we are going to do, then we close the chapter on the urinary system, is the mechanism of micturation. In fact, when we say micturation is the physiological process by which urine is expelled from the urinary bladder through the urethra to the outside of your body. So when we say micturation, this is a physiological process by which urine is expelled from the urinary bladder through the urethra to the outside of your body. Okay, that's micturation. Okay, now, micturation actually starts from the kidneys, because you see, the kidneys are the ones that are helping to form the urine. So when the kidneys filter the blood to produce the urine, okay, which flows via, so it produces the urine, it flows to the renal pyramid, to the renal papilla, to the minor callus, to the major callus, to the renal pelvis, to the ureters, from the ureters to the urinary bladder, from the urinary bladder to the to the urethra. Okay, so once it hits into the urinary bladder, that is where the micturition process is now going to come in. You remember we mentioned that the urinary bladder is a hollow muscular organ, which is dominated by what we call the detrusor muscles. And this detrusor muscle stores the urinary bladder helps to store urine until the urge to void arises. Okay, now usually as the bladder fills about 300 to 400 mils, we have stretch receptors in the bladder wall which sends afferent signals to the spinal cord, that is S2 and S4. Now we have what we call the spinal micturition reflex center, which is located in the sacral spinal cord, the S2, the S4. This center is what actually initiates involuntary detrusor contraction and internal sphincter relaxation via the parasympathetic fibers. So in effect, the internal urethral sphincter, which is made up of smooth muscles at the bladder neck, it relaxes involuntarily during micturition. And then again, external urethral sphincter, which is made up of skeletal muscles under voluntary control. Okay, control via the pudendal nerves, it allows conscious control of urination. Okay, then the urethra, which is also the passageway through which the urine exits the bladder, differ in length in terms of males and then females. Okay, then we have the cerebral or pontine control, whereby the higher brain centers, we have the micturition centers in the brain, okay, and the cortex. They modulate the reflex. They allow voluntary initiation or suppression of urination depending on the social context. So now these are the structures that are involved in micturition. So now we are going to look at micturition mechanism itself. In fact, the mechanism of micturition involves two phases. Okay, we have the storage phase, whereby the urine have to be stored or kept in the urinary bladder, and we have what we call the voiding stage or phase. That stage is the micturition phase. Okay, so the mechanism of or physiology of micturition is in two phases. We have the storage phase and we have the voiding phase. The voiding phase is the micturition. Okay, so let's start with the storage phase. Okay, let's start with the storage phase. Now, the detrusor muscles in the storage phase, the detrusor muscle, because we want to store urine. So what will happen is that the muscles of the urinary bladder do not have to contract, they have to relax. So in the storage phase, the detrusor muscles will have to relax. So the smooth muscles in the walls of the urinary bladder would remain relaxed in the storage phase. Once they relax, the internal sphincter and then the external urethral sphincter, they would rather be the ones to contract. And anytime the internal urethral sphincter and the external urethral sphincter contracts, is that okay? You hold urine or you store urine. It means that you are not urinating. So if you are keeping urine, you are contracting your sphincters. You are contracting them when you are keeping urine. So the more you are keeping urine, the more you are contracting your sphincters. Okay. So if you in the phase where you are storing urine, your urinary bladder, the detrusor muscles would relax. Then the sphincters, the external sphincter, and then the internal sphincter and the external sphincter would rather contract for you to hold the urine. Now, the key receptors that enables this to happen is that in your urinary bladder, we have some receptors. We have the beta-3 adrenergic receptors. Okay, the beta-3 adrenergic receptors in the detrusor muscles. Okay, these beta-3 adrenergic receptors, they are activated by sympathetic nerves via the norepinephrine release, and they cause what we call the relaxation of the detrusor muscles and allows the bladder to fill sufficiently. Then we also have the alpha-1 adrenergic receptors in the internal urethral sphincter. These alpha-1 adrenergic receptors, they also stimulate what we call sympathetic nerves, and they cause contraction of the internal sphincters, and that is going to prevent leakage. Is that okay? The alpha-1 adrenergic receptors in the internal urethral sphincter, okay, they are stimulated by the sympathetic nerves, and they cause contraction of the internal sphincter to prevent leakage. Then we have the nicotinic receptors. Okay, the nicotinic receptors are in the external sphincter of the somatic external sphincter, and they are of somatic control via the pudendal nerves. Okay, so under voluntary control, when you want to initiate urination, they are activated and they keep the external sphincter. If you want to hold urine, okay, they are activated and then they keep the external sphincter contracting. Okay, and in that phase, you are holding urine so that there wouldn't be any leakage. Okay, so anytime you are holding urine, it means that the adrenergic receptors have been activated by the sympathetic nerves. When the adrenergic receptors are activated by sympathetic nerves, it is activated by the norepinephrine. So that will cause relaxation of the detrusor muscles. Okay, and that relaxation will enable the bladder to fill to capacity. Okay, then the alpha adrenergic receptors, okay, in the internal urethral sphincter, they are also stimulated by the sympathetic nerves. But then when they are stimulated by the sympathetic nerve, they cause contraction because they are stimulated via what we call adrenaline release. So they will cause what? Contraction. It will cause contraction of the internal sphincter, and that will what? Prevent leakage. Okay, then the nicotinic receptors in the external sphincter, they are under somatic control via the pudendal nerves. Okay, and for the pudendal nerves, once they are activated, they cause what we call contraction, and in that situation you are storing the urine. Okay, good. Then we have the voiding phase where you are urinating, you want to rather urinate. In that sense, then it means that we have another receptor within the detrusor muscles. We call them the muscarinic receptors. The muscarinic receptors, we also call them the M3 or M3 subtype receptors. They are stimulated by the parasympathetic nerve via acetylcholine release, and they cause contraction of the detrusor muscles and initiate urination. Is that okay? And then again, there is going to be inhibition of the alpha receptors, and that would also lead to relaxation of the internal sphincter. Now, there is also going to be inhibition of the nicotinic receptors, which would cause relaxation of the external sphincter, and at the end, that will allow the sphincter to open. Then the person now urinates. This mechanism is what we call the mechanism of micturation. Mechanism of micturation. Okay, so in summary, it has two phases: the storage phase and then the voiding phase. The voiding phase is the micturation. With the storage phase, the summary is that the smooth muscles or the detrusor muscles have to relax. Then the internal sphincter and the external sphincter have to contract. In that case, you are storing the urine. Okay. Now, if you want to void, then it means that the detrusor muscles have to what? Have to contract. Okay. Then in the end, the sphincters have to now what? Relax. Then in that case, there will be urine leakage. Then you urinate. That's the summary of the whole micturation process. That is basically it. Okay. Good. Now, so basically, we've come to an end of the urinary system anatomy. Now, the clinical significance or the importance of studying urinary system is that it will help to guide you in safe catheterization techniques, because as a medical person, there will come a time that you may have to assist or perform urinary catheterization or urethral catheterization. Okay, so your knowledge on the urinary system, okay, and then the bladder anatomy, ureter anatomy, all would help you to appreciate the whole process. Okay, so that you'll be able to minimize risks, trauma, and also be able to appreciate why you have to, conduct these procedures in a sterile environment. Okay, now it also enables accurate diagnosis and treatment. Differentiating between the upper and lower urinary tract, okay, depends on the understanding of the renal pelvis, the ureters, and then the bladder structure. Okay, it also supports surgical and procedural safety. Your awareness of the urethral and bladder anatomy prevents injury during surgeries like hysterectomy, prostatectomy, and then stent placement. It also improves fluid and electrolyte management. So your knowledge on urinary system anatomy, okay, urinary system anatomy, especially the nephrons, is that okay? Your awareness of it, okay, would aid in your adjusting to fluid therapy during renal failure or edema to avoid overload or dehydration. It also enhances interpretation of diagnosis tests, okay, because now you can now understand creatinine. If creatinine is accumulating in the blood, you would appreciate what it means and the impression it can give you about the functions of the kidney. If blood urea nitrogen is accumulating, it will give you an impression of what is happening in the kidney. If glomerular filtration rate is high, if glomerular filtration rate is low, it will also tell you something about the kidneys. Good. And then it also helps you to appreciate the need for monitoring intake and output. The reason why you as a health professional, for some conditions, you have to keep an eye on intake and output monitoring. It would also help you to understand and support management of neurological and systemic conditions like issues relating to micturition reflex, renal vascular connections in key conditions like neurogenic bladder, diabetes, or hypertension. Okay, so this is what we were talking about, that it will help you to appreciate catheterization techniques, because catheterization techniques, you appreciate it when you are going to do catheterization for males and when you are going to do catheterization for females, because you now know the differences between their ureters. Okay, and the reason why sometimes during catheterization we may have to inflate the balloon. Okay, we have a port for inflating balloon on a different side. Okay, good. So basically that is the application. Okay, and also it will help you to understand certain medications, the action of certain medications like renin-angiotensin converting enzyme inhibitors. Okay, angiotensin converting enzyme, ACE inhibitors, and then the ARBs. So help you to appreciate their mechanism. Okay, good. And then it will also help you to improve on your postoperative urinary care, especially when you are dealing with post-op cases, patients on indwelling catheters and all that. It will help you to appreciate the care, help you in also monitoring intake and output and making some clinical decisions with intake and output. Is that okay? It will also help you to appreciate the reason why in some cases or in some patients you may have to encourage patients on the need for adequate hydration. Is that okay? Good. So basically that is it for the urinary system. Now the last thing we are going to look at is, let's look at some structural disorders and conditions that affect the kidney anatomy. Now we have a condition we call glomerulonephritis, which is an inflammation of the glomerulus. We have nephrotic syndrome, a condition whereby the glomerular filtration membrane or the glomerulus gets damaged. We have renal failure. We have polycystic kidney disease. Okay, a condition where we have formation of multiple fluid-filled cysts in the kidneys. Multiple fluid-filled cysts. This condition. Multiple fluid-filled cysts in the kidney. That is what we call polycystic kidney disease, PKD. We have hydronephrosis, a condition where we have swelling of the kidney due to urine buildup. We have renal agenesis. This is hydronephrosis. Okay, this is hydronephrosis. The kidney swells up. Okay, swelling of the kidney due to urine buildup. We have renal agenesis, where there is absence of one or both kidneys. There are times someone can be born with just one kidney, or someone can be born without any kidney. There is absence of kidney. We call that renal agenesis. Then we can have what we call the horseshoe kidney. This is the horseshoe kidney. Okay, so at the poles of the kidney, you see that there is fusion of the two kidneys at the lower poles. So at the lower poles, you see fusion of the two kidneys at the lower poles. This is what we call the horseshoe kidney. Then we have duplex kidney, whereby there is a duplicated collecting system. Duplex kidney. There is a duplicated collecting system. So the kidney with two ureters or renal pelvis. Okay, sometimes you can have a kidney with 2 ureters or a kidney with 2 renal pelvis. Okay, that is what we call the duplex kidney. Then we have nephrotosis or floating kidney, whereby there is an abnormally downward displacement of the kidney. We have medullary spongy kidney, where there is a cystic dilatation of the collecting ducts in the renal tubule. We have kidney stones or nephrolithiasis. Okay, we have chronic pyelonephritis. We have renal artery stenosis, renal infarction, where there is death of kidney tissue due to loss of blood supply. We have renal cell carcinoma, renal hypoplasia, and renal dysplasia. Okay, so these are structural disorders and conditions that can affect the kidney anatomy. Okay, so this brings us to the end of the urinary system. In Leviticus chapter 3 verse 10, and it is stated there that the two kidneys and the fats that are on them, which is by the lions, and the cover on the liver with the kidneys he shall take away. All right, so this brings us to the end of the urinary system anatomy. All right, thank you so much. All the best.